Welcome to Plenary Session 2, Lifestyle Impacts on Immunotherapy. We have a terrific lineup of speakers, and here is Dr. Iyengar to introduce our first speaker.
I have the honor of introducing Drs. McQuade and Dr. Norian. We'll start with Dr. McQuade. Dr. McQuade is an assistant professor and physician scientist in melanoma medical oncology at the MD Anderson Cancer Center. Her research focuses on how lifestyle factors influence melanoma biology and the anti-tumor immune response with the goal of developing novel strategies to improve outcomes in melanoma. We're excited to have Dr. McQuade with us today to share an update on her work. And with that, Dr. McQuade, I'll hand it over to you.
Okay, thank you, Neil, for the kind introduction, and to the organizers for inviting me today. Today, I'm going to be discussing our work looking at the impact of host metabolic phenotype, and specifically obesity, on response to immunotherapy and on tumor metabolism. And these are my disclosures. And so given that I'm the first one in this session, I thought that I would start by just introducing a bit immune checkpoint inhibitors and really the role of immunotherapy in cancer. So everybody has now heard of immune checkpoint inhibitors. These have led to 2 Nobel Prizes, and they have really revolutionized the treatment of many malignancies. So immune checkpoint inhibitors were actually first approved in metastatic melanoma. Now, metastatic melanoma is a disease that has traditionally had a very dismal prognosis, a prognosis actually similar to that of pancreatic cancer, with long-term survival less than 10%. With immune checkpoint inhibitors, we've improved that overall survival from less than 10% to now 50 to 60% of our patients living 5 years. What we get very excited about, of course, when we look at immune checkpoint inhibitors is this tail of the curve. So patients that will have disease that is completely eradicated can come off of immune checkpoint inhibitors and live for many years disease-free, potentially even cured. We also have decent response rates, and really in melanoma, one of the highest of any malignancies. However, 50% of our patients, even with metastatic melanoma, fail to achieve any benefit whatsoever from immune checkpoint inhibitors. So what are these drugs? So cancer, in a way, is a disease of immune escape. Our immune system, of course, goes around and surveils for viruses and bacteria, but also looks for precancerous and cancerous cells. And the way that it recognizes that these tumor cells are different and need to be eradicated is because tumor cells, when they have mutations, now express different antigens, different proteins on their cell surface that can be recognized by the T cells. However, tumor cells basically evade the immune system by expressing these immune checkpoints that essentially tell the immune system, go away, I'm just a normal cell. So the discovery that led to the Nobel Prize was the discovery of the immune checkpoint inhibitors, and then the further work showing that if you now give an antibody that blocks these immune checkpoints, then you're able to restore this balance and essentially take the brakes off of the immune system, now allowing the immune system to come in, recognize, and kill a cancer cell. And so what factors determine tumor response versus resistance? There's, of course, a ton of research around this. So really we think about usually tumor intrinsic pathways, namely oncogenic signaling pathways, or we think about tumor extrinsic, the immune microenvironment. And so is this a tumor that is hot and inflamed with lots of T cells in there trying to do their job? One factor, though, that really had not been well studied before was this dimension of the host. So what about the role of things such as age and sex on anti-tumor immunity in the setting of immune checkpoint blockade? Or then what my patients ask me when I tell them that your tumor has a 50% chance of responding to immunotherapy is, Doc, is there something else that I can be doing? And so what about the role of modifiable factors such as obesity? And we'll hear more, diet, exercise, and what happens when we get into stress, sleep? Are there things that our patients can be doing that influence outcomes with immune checkpoint blockade? And then we will have one talk in this session on the microbiome. Really, the microbiome, I think, has led to a lot of the work that we do in energy balance becoming relevant again as really an integrator of many of these factors and how this now impacts immunity. So there's, of course, a strong rationale to look at these host factors. Again, had not been well studied in the context of immune checkpoint blockade, but we do know that many of these factors influence our ability to fend off viruses, our vaccine response, our outcomes with COVID. So age, the microbiome, genetics, obesity, viral infections, all of these things can influence our overall immune tone, our overall set point between inflammation and immune tolerance. Now, we became interested in this in melanoma for a very specific reason, and that is because of the relationship between obesity and higher levels of insulin and insulin-like growth factor. So this is, of course, just one mechanism by which obesity can promote carcinogenesis. But IGF-1 is actually a critical node in melanoma biology. So 50% of melanomas have a mutation in the BRAF oncogene. This is a gene, it's part of our normal cell signaling pathways that tells our cells to grow and divide. And in these 50% of melanomas that have this BRAF mutation, this pathway is constitutively activated. Now, upstream from these pathways, as well as these other oncogenic signaling pathways, such as the PI3 kinase pathway, live receptor tyrosine kinases that are responsive to growth factors such as insulin and IGF-1R. And work from my colleagues at MD Anderson has previously shown that IGF-1 could activate the PI3 kinase pathway and lead to resistance to both targeted and immunotherapy in melanoma. So here we actually see that in patients that have the PI3 kinase pathway activated by PTEN loss, we actually have immune exclusion in those specific regions of their tumor. Now, again, we also know that obesity can negatively impact adaptive immunity by the state of chronic inflammation, which has been termed inflammaging. And so we hypothesized that obesity would lead to therapeutic resistance in melanoma through activation of the PI3 kinase pathway and impaired immune response. And so we first studied this in mice. In a very well-validated model, we took male C57 black 6 mice, fed them either a 10% fat diet or a 60% fat diet for 15 weeks, had reliably higher weights, IGF-1, glucose. And when we now inject these tumors with molecularly relevant BRAF mutated melanoma cell lines, then we see increased tumor growth. And when we look at the molecular level, then again, we see that this host metabolic phenotype impacts the tumor oncogenic signaling pathways. And so by both reverse phase protein array as well as by IHC, we see activation of the PI3 kinase pathway. Here is measured by phospho-S6. And so we next looked at this in patients. And so this is work that we published in Lancet Oncology in 2018. This is a study of over 1,900 patients in 6 independent cohorts looking at the relationship between body mass index and outcomes with either targeted therapy, so BRAF MEK-directed therapy, immunotherapy, either PD-1 or ipilimumab, anti-CTLA-4, and then 2 cohorts of patients treated with chemotherapy, which is not very effective with this disease. And so we first started with targeted therapy. And what we see is that to our surprise, obesity here in blue was associated with significantly improved both overall survival that I'm showing here, but also progression-free survival, as well as overall response rate. We see the same thing when we look at patients treated with bemirathen and cobimetinib. And here now, when we look at patients treated with immunotherapy, we see that our obese patients have improved outcomes with PD-1. We also see the same thing with ipilimumab. Now, so we then, of course, asked the question, is this just generally prognostic? And when we look at patients treated with chemotherapy, there's actually no difference in outcomes by BMI status. So again, we saw very consistent results between the two different targeted therapies and between the immune therapies. Nothing with chemotherapy, but basically a 40% decreased risk of death in our obese patients treated with either targeted or immune therapy. And so it seems that obesity was specifically predictive of response to targeted immune therapy and not generally prognostic. And I think that this is an important distinction because we have seen before that you can have this obesity paradox in diseases. And a lot of times it comes down to our obese patients being better able to withstand treatments, being less likely to have experienced significant weight loss or malnourishment into therapy. And we see this across surgery as well as across different treatments. So we, of course, looked at different prognostic factors. And indeed, our patients that have higher BMI were more likely to be males, older, slightly more likely to have more advanced stage disease. But the associations remain significant after adjusting for age, sex, stage, LDH, as well as performance status. We also looked into concomitant medications. Our patients with higher BMI are more likely to be taking medications for metabolic syndrome. These medications in and of themselves may have anti-cancer activity, but the associations remain significant after we added this into the model. In terms of treatment tolerance, again, we did not observe this with chemotherapy. Chemotherapy being the treatment that would be more likely to lead to weight loss. We see no difference with chemotherapy, and we saw no decreased risk of any adverse events with immunotargeted therapy in our obese patients. We also had PK data from the vemurafenib cobimetinib trial, no difference in drug exposure. And finally, as far as metabolic reserve, our patients here were not cachectic. They were not underweight. 65% of these patients were overweight or obese, mirroring the US population, and less than 2% of patients were underweight, and we excluded those. So we, of course, have to ask the question, is BMI the right exposure? We all know that BMI is an imperfect surrogate of adiposity. Betty Kahn has previously done some really nice work looking at body composition and outcomes, where in colorectal cancer, what we see on this line curve is basically, it's not truly an obesity paradox, it's an overweight paradox. This BMI of about 25 to 30 seems to be the sweet spot in terms of survival. And when you look at body composition here, you see that those are the patients that are actually less likely to be sarcopenic and then don't have too much adiposity. So again, kind of that sweet spot around BMI of 25 to 30. Now, in contrast with melanoma, we actually see a dose response. And so melanoma on the top, colorectal cancer on the bottom, what we see with melanoma is all the way through the range of prevalent BMIs up to a BMI of about 40. The more obese patients are, the better they do. We also asked the question of, is there true metabolic dysfunction in these obese patients? And yes, BMI was correlated with higher levels of C-peptide, IGF-1, and leptin, indicating that these patients do have metabolic dysfunction. And so with this dose response curve, although we are actively working on doing body composition analysis, it seems less likely that adiposity is classified at a BMI of 40. And we do see that BMI does correlate with these blood-based biomarkers of adiposity and melanoma. So this finding has actually been validated by many, but not all studies, as Dr. Norian will get into, again, some of the differences in different cancers and in different ways of looking at this. So this paper on the left, and I apologize if the reference came off, so Dr. Wong, so again, here now looking at patients treated with immune checkpoint blockade across many cancer types, we see improved progression-free survival in the obese, and this was actually a meta-analysis that was published in 2020, again, seeing improved overall as well as progression-free survival overall across many studies. So how could obesity possibly be good for immunotherapy response? And so in this paper published in Nature Medicine, they see is that similar to what we've seen when you inject mice with melanoma tumors, you increase melanoma tumor growth in the obese. They noticed then that obesity was inducing upregulation of CD8, PD1 expression, and then leading to functional exhaustion. And again, why would this be good? Well, this PD1 upregulation could provide a target for anti-PD1 based immunotherapy. So paradoxically rendering them more vulnerable to treatment with immune checkpoint blockade. Now, in those studies, they looked at the immunological correlates of obesity in limited human samples. And so here, we also see higher PD1 T cells in the obese and circulating healthy volunteers, lower CD8 infiltrates in colorectal tumors of obese individuals. And they had a limited subset of the TCGA that had BMI available. When they restricted it to patients aged greater than 60, then they see higher expression of multiple immune checkpoints in the obese tumors. So we decided to dive into the molecular and immune correlates of obesity in human melanoma much deeper. And what we did is we took patients, divided them into high versus low BMI, and then looked at DNA profiles, RNA-seq, RPPA, as well as performing metabolomics by multiple different methods, and then directly looking at immunohistochemistry of several immune pathways, and finally doing an analysis of the stool microbiome. And so what we see when we look at immune infiltrates in our hands, so this is the MD Anderson cohort and the Melanoma Institute of Australia cohort. And the red is the overweight obese tumors. What we see is that kind of a hint of a skewing perhaps towards higher infiltrates or higher expression of these checkpoints in the overweight or obese. Although visually it looks like that, I can tell you that the medians and the means are about exactly equal, and it really is some outliers in the obese that seems to be skewing this. But really no statistically or clinically meaningful difference in the immune infiltrates between the obese and the non-obese. So we next were able to get BMI on many more patients from the melanoma TCGA, and then restricted this analysis to those with regionally metastatic specimens to improve the homogeneity of the cohort. And what we see here is that we actually see no differences in mutations or copy number variations. Critically, also no difference in tumor mutation burden. Tumor mutation burden has been very well validated as one of the biomarkers that predicts response to immune checkpoint blockade. No difference in tumor mutation burden by BMI. And again, we do see higher serum IGF-1 in the obese, but contrary to our hypothesis, the PI3 kinase pathway is not activated in the obese. So we further looked at RNA sequencing, both from the TCGA cohort, but also gene expression profiling from 3 other independent cohorts. And the way that we did this was by first batch correcting the different cohorts so that we could combine and analyze together. And so this map is comparing gene set enrichment analysis of overweight obese tumors compared to normal BMI tumors. And here, red means upregulated in the obese and blue means downregulated. And what we actually saw was downregulation, most strongly of oxidative phosphorylation across the entire data set, and then also when we controlled for sex data set as well as tissue type. We also see downregulation of other metabolic pathways as well, including myogenesis, lipolysis, and adipogenesis. The oxidative phosphorylation finding really caught our attention because oxidative phosphorylation has previously been shown to drive resistance to immune checkpoint blockade. And in general, metabolic phenotypes of the tumor actually impacting the functionality of immune cells by the more metabolically active tumors, essentially outcompeting the immune cells for nutrients. So we then asked, we see downregulation of OXPHOS and glycolysis. Is there compensatory upregulation of an alternative metabolic pathway? And here, when we take a step back, we do gene set enrichment analysis that now looking at all of the KEGG metabolic pathways, we actually see that the tumors from the overweight obese individuals seem to be more globally metabolically quiescent. We had fresh frozen specimens from some of the tumors that contributed to the melanoma TCGA, and we measured TCA cycle metabolites directly by mass spec. What we see in the overweight obese is decreased levels of citrate and succinate. I think more importantly, though, we then worked with Fred Delgoff and Yana Najjar at the University of Pittsburgh, and basically did ex vivo measures of oxidative phosphorylation and glycolysis through the seahorse extracellular flux assays. And so essentially what they do is they take tumors, they then sort out these CD45 positive immunocytes. We now have single cell suspension of tumor cells, and then stimulate those and basically then measure oxidative phosphorylation and glycolysis. And so we won't go into the details of exactly how this works, but what we see is that here, this is oxidative phosphorylation of these tumors, which is lower in the overweight obese compared to normal BMI. And so then we took it a step further and we actually looked at this as well in patient-derived melanoma cell lines. And so these are cell lines from our patients that we keep at very low passages. My colleague Wade Pang, who's now at the University of Houston, had actually measured oxidative phosphorylation and glycolysis in 17 of these cell lines. And what we see is that in those cell lines derived from overweight obese individuals, we see lower oxidative phosphorylation and glycolysis. And so we actually see this conserved ex vivo. And finally, we also looked at the gut microbiome. So the gut microbiome has, of course, been shown to impact response to immunotherapy in terms of both specific taxa that might be associated with response, but also one of the things we look at is the diversity of the gut microbiome, meaning how many different types of bacteria are present, indicating the overall richness and health status of the gut microbiome. And what we see here actually is that our patients with higher BMI, consistent with some other literature, tend to have lower diversity of the gut microbiome. Now, diversity of the gut microbiome, this would be in the opposite direction from what would be beneficial. So it does not seem that the obesity link is via the gut microbiome in terms of response to immune checkpoint blockade. And then here on the right, a PCOA plot, basically showing that there's no large structural differences between the gut microbiome of obese versus non-obese. So in conclusion, obesity in some studies, including ours and not all, is associated with improved outcomes with targeted immune therapy and melanoma. In melanoma, obesity is associated with downregulation of multiple metabolic pathways, which have been shown to mediate resistance to these therapies. The conservation ex vivo suggests a possible epigenetic mediator, which needs to be further investigated, as well as the impact of obesity on immune cell metabolism, what I showed you was tumor cell metabolism, and immune cell function. So again, beyond immunohistochemistry and RNA sequencing. So I'd like to thank the members of my lab. Andy Hahn is a very talented medical oncology fellow who has been involved in all of this human work, and then the rest of my team that does our mouse work, a really great computational biology group. Mike Davis, my mentor initially, and colleague on this work, urodynamic nutritional epidemiologist, and then Jen Wargo with all of the microbiome work, Lorenzo Cohen and Kane Young, initial mentors with the preclinical work. The Seahorse was done by Greg Delgoff down in Achar and Willie Payne, and then external collaborators around the world that have actually contributed data to these analyses. And finally, my funders, and just a plug, that always looking for good stuff. Thank you.
Next, I'd like to introduce Dr. Norian. Dr. Norian is an associate professor in the Department of Nutrition Sciences at the University of Alabama at Birmingham. Her lab is currently focused on two main areas: defining the mechanisms of tumor-induced immune dysfunction and understanding how diet and obesity alter the quality and magnitude of anti-tumor immune responses and immunotherapy outcomes. Dr. Norian.
Thank you. So I'll be talking to you today, a little different emphasis than what Dr. McQuade just covered. So my lab's research on identifying drivers of obesity-associated immunotherapy resistance. And I have this image here, which is a very famous optical illusion of the illustrator's young wife and his much older and less attractive mother-in-law, and I'll come back to that at the end. So when I began this project, we really were asking what we thought was a very simple question, which was, does host obesity impair immunotherapy outcomes? And the reason for studying this is because although obesity has been a human condition for a long, long time, the image here is a picture of a statuette known as the Venus of Willendorf. She dates from 23,000 BC. Obesity has taken on new significance due to its prevalence. In the United States, it's estimated that about 39% of adults have obesity as measured by their body mass index, or BMI. So obesity is also known to increase the risk of developing 13 different types of cancer. Among these is kidney or renal cell cancer, which is what I'll be focusing on today. You'll notice on this list, absent is melanoma, the tumor type that Dr. McQuade had just been discussing. So kidney cancer is one of the top 10 cancers for both men and women, and renal cell carcinoma, or RCC, is the most common subtype of kidney cancer. Obesity is one of the three major predisposing factors for RCC, and for this presentation and this line of research, the fact that RCC is immunogenic is really important, and I'll get into the immunogenicity of the tumor a little bit on the next few slides. So for a long time, immunotherapy was really the only known curative treatment for advanced kidney cancer, and that came in the form of high-dose IL-2 or interferon alpha. High-dose IL-2 in particular was associated with very substantial toxicities that contraindicated its use in many patients. Then in 2015, anti-PD-1 was approved, and in 2018, the combination of anti-PD-1 and anti-CTLA-4 was also approved. And so immunogenic tumors activate protective anti-tumor immunity. So what I'm showing here in this diagram is just a solid tumor that is infiltrated by different types of leukocytes, dendritic cells shown in blue and T cells shown in gray. We know that as tumors progress, there's always tumor cell death occurring. And what results from this is that dendritic cells are able to take up portions of these dying tumor cells and carry them to the tumor draining lymph node, where they can present peptides derived from these tumor proteins to naive CD4 or CD8 T cells. This causes the T cells to undergo what immunologists refer to as clonal expansion. So we start with one cell, we end up with many, all specific for the same tumor antigen, and these cells become activated, they leave this lymph node, they traffic systemically, where they can follow chemokine gradients back to the tumor. And if everything works well, they can bring about tumor cell death. So we know that this doesn't happen often without therapeutic intervention. And as Dr. McQuade mentioned previously, we know that a major pathway that prevents T cells from killing tumors is PD-1 engagement with PD-L1. And so many studies have shown that if we block this interaction between PD-1 and PD-L1 with antibodies, that the T cells in the tumor microenvironment can retain their function and help to control tumors by causing tumor cell death. So these immune checkpoint blockade agents or immune checkpoint inhibitors have really changed the landscape of treatment for advanced cancer patients. But although anti-PD-1 can improve the intratumoral T cell response, there are many factors that limit its efficacy. And one that I'll be talking about today specifically are immunosuppressive cell populations, such as myeloid-derived suppressor cells within the tumor environment. So when we started looking at this, we wanted to know in renal cancer patients, did obesity impair anti-PD-1 outcomes? And the reason for this, as Dr. McQuade said, there have been studies prior to ours showing that obesity was actually beneficial, particularly in melanoma patients, as she had just discussed, in terms of immune checkpoint outcomes. And so we were interested to know if the same type of response was seen in an obesity-associated cancer, renal cancer. So we started here by taking fresh tumors from kidney cancer patients. We have them divided out here into non-obese individuals, BMI less than 30, or individuals with obesity who have a BMI equal to or greater than 30. And what you can see is that in the tumors from individuals with obesity, we have a trending but non-significant reduction in the frequency of activated CD8 tumor infiltrating lymphocytes or TILs. When we looked specifically at PD-1 positive CD8 TIL subpopulations, we found that opposite to what has been described in melanoma, we actually have fewer PD-1 positive CD8 TILs in the tumors from our renal cancer patients when those patients have obesity. And it's not a reduction in the PD-1 intermediate cells, it's actually a reduction in the PD-1 high cells within the tumor that is causing the significant decrease. Interestingly, if we look in circulation at CD8 T cells in terms of their PD-1 expression from either RCC patients or tumor-free healthy volunteers, we see no increase in the frequency of PD-1 positive CD8 T cells as has been described by other groups. So we took another look at this, looking at plasma leptin, which we know is increased with increasing BMI. And we asked, okay, so as BMI increases, if we look at these peripheral blood T cells, what happens with their the frequency of PD-1 high T cells, and we see that it decreases. We also see that as plasma leptin increases, we see a decrease in PD-1 high CD8 T cells. So again, very interesting, but the opposite of what has been seen in melanoma patients. So as Dr. McQuade mentioned, there's this hypothesis out there that perhaps in melanoma, where they had seen an increase in PD-1 positive CD8 T cells within tumors, that provided more target for anti-PD-1. And so that was a partial explanation for why immune checkpoint had better outcomes in patients with obesity. So then it was very important for us to look at our renal cancer patients and ask a similar question. So what we did was we took all the renal cancer patients from UAB hospitals, as well as patients from the University of Iowa hospitals and clinics. And these were patients who received only anti-PD-1, so no combination of anti-PD-1 with anti-CTLA-4 or any other agent. And these were patients who received anti-PD-1 as standard of care, so not on clinical trials. And what we found, again, was opposite of what had been reported in melanoma. So we see that for both progression-free survival and overall survival, the individuals who had obesity actually did much worse over time than did the individuals who either had a normal weight or overweight status. But I think the important thing to look at here is not just this overall trend, but the fact that some patients with obesity actually do respond very well to anti-PD-1. And so that led us to look back at all the studies that have been published so far. And what is really obvious is that in all of these reports on BMI-defined obesity and cancer immunotherapy outcomes, the individual responses of patients vary, and they include both therapy responders and non-responders, but these vary to different extents depending on the cohort or cohorts of patients that have been studied. And so this we thought was fascinating and led us to ask a slightly different question, which is, well, what are the drivers of this obesity-associated immunotherapy resistance? And to do this, we used a model of diet-induced obesity that we have used previously in our lab. So we have mice, BALB-C here, that are randomized to either standard chow or a high-fat diet for 20 weeks prior to use. What we're looking at here on this slide, each dot is the weight of an individual mouse after being on diet for 20 weeks. So you can see there's a range of weights within the cohort that has been on standard chow. And within the cohort on high-fat diet, you can see there's a remarkable variation in body weights. And so clearly we can't lump these mice in with these and refer to them all as obese. So what we have decided to do in my lab is to objectively define diet-induced obesity in mice as any animal who has a body weight greater than the lean mean, so the standard chow mean plus 3 standard deviations. So these animals are diet-induced obese. Animals on high-fat diet that have a body weight within 1 standard deviation of the lean mean cohort, the lean cohort mean, we refer to those as obese resistant. So if we look at all 3 of these groups, the lean, the obese resistant, and the diet-induced obese mice, you can see it's only the DIO mice that have these classic hallmarks of obesity, so elevated leptin and insulin systemically. So then we combined this model of diet-induced obesity with an intrarenal tumor challenge to have a preclinical model of renal cancer. And so what we do in this model is we have renal carcinoma cells called RANCA that we engineered to express luciferase. We put them directly into the kidney on day 0. And then between day 7 to 28, we monitor tumor outgrowth and immune responses to tumors in a variety of ways. So this is a very aggressive model. So histology shows us that by day 7 post-challenge, you can see in the purple here, areas of tumor growth that are evident. By day 14, we have large areas of the kidney that have been completely taken over by tumor. And then because our cells are transduced with luciferase, we can use bioluminescent imaging to track tumor growth over time. So it's a pretty nice model. So we then wanted to use this model to study the effects of obesity on anti-tumor immunity as well as immunotherapy outcomes. And so what we did here was in mice that had not received any therapy, similar to what we did with our renal cancer patients, we took tumors, renal tumors from these mice, and we looked at the frequency of activated CD8 TILs or tumor infiltrating lymphocytes, and we found a trending decrease. Then when we looked at PD-1 positive CD8 TILs in these renal tumors, we found something that reflected just what we had seen in our renal cancer patients. So in the mice with diet-induced obesity, they had a significant reduction in these PD-1 high CD8 TILs in their tumor-bearing kidneys. Interestingly, though, when we look at the function of these PD-1 high cells, if you look here, this is PD-1 expression on the x-axis versus perforin on the y-axis here, you can see that in both lean mice and obese mice, the PD-1 high cells in this model are still functional. So they retain perforin expression and interferon gamma production. So we then wanted to immediately ask the question, well, what happens if we give anti-PD-1 to these mice? But this tumor model is very aggressive and it's highly resistant to anti-PD-1 monotherapy. So we had to back up and devise a combinatorial therapy that would basically increase the frequency of CD8 T cells that are activated in response to tumor growth, and then come back with an anti-PD-1. And so the therapy that we decided to use is an adenovirus encoding trail plus CpG. And this slide just is a visual explanation of how this works. So adenovirus encoding trail is a protein that's known to induce apoptosis in tumor cells. And what this does basically is increase the material that's available for dendritic cells to take up. And then the CpG portion of the therapy actually helps trigger dendritic cell maturation. So when these dendritic cells get into the tumor draining lymph nodes, they are better able to activate CD8 T cells. We have more clonal expansion and more activated T cells that can get back into the tumors and bring about the destruction of these tumors. So in lean mice, we started by looking at the efficacy of this therapy. And so I'm showing you tumor weights on the y-axis over here versus different treatment groups along the x-axis. And you can see in mice that don't receive any therapy, they develop these large, very bulky renal tumors. This red dotted line here shows you the weight of a tumor-free kidney. And all these dots are individual mice. You can see if we give anti-PD-1 alone by day 28 after tumor challenge, it really, as I said, is not effective at controlling tumor growth in this model. But if we give ADTrail plus CpG alone, we get a 55% reduction in tumor burden versus mice that receive no therapy. And if we combine ADTrail CpG with anti-PD-1, we get a 75% reduction in tumor burden. And we use this 75% reduction as a responder threshold because you can see that we have some mice here, even with this combination therapy, that respond well, and we have some mice that do not. Okay, so now we have everything in place. So we were able to ask the question, if we have mice on high-fat diet who are DIO, or obese resistant, or mice on a chow diet that are lean, and we give ADTrail CpG on day 7, follow it up with anti-PD-1, what happens in terms of therapeutic efficacy? And what I'm showing here is that in lean mice, again, as I showed you previously, this combination therapy works very well. So we have no therapy mice, showing these large tumors, renal tumors growing out. Lean mice that receive therapy, we have this nice reduction in tumor burden. And if we use that 75% reduction to objectively define responders and non-responders, we see that 73% of these lean mice respond to this therapy. In contrast, for the DIO mice, you can see that the no therapy mice, again, have these large tumors that grow out. The therapy treated mice have a trending but non-significant overall reduction in tumor burden. But if you look at the response rate, you can see it's only 44%. So is this difference in response due to the change in diet from the chow diet to the high-fat diet? And this is where we use our obese resistant mice that have also been on the same high-fat diet for 20 weeks. And we see that the obese resistant mice actually respond just as well as lean mice on chow diet to this therapy, giving us an overall 73% response rate. So it's really the obesity and not the high-fat diet administration that's diminishing treatment efficacy in this renal cancer model. So we next wanted to determine, so what is accounting for these poor outcomes in the mice with diet-induced obesity? So we took a step back, and what we decided to do was to do an unbiased immunogenetic profiling of tumors from lean versus obese mice. And we included all groups. So we had no therapy, therapy responders, and therapy non-responders. And you can see from the unbiased hierarchical clustering here, we really have three groups across the top. So this group here contains both lean and obese mice that responded to therapy. This central group here includes DIO mice that had no therapy, lean mice that received no therapy, and DIO mice that did not respond to therapy. And then interestingly, but not the focus of the talk today, we had lean mice who did not respond to therapy that were off on their own. So then we wanted to look more closely at the DIO mice and their gene expression signatures for mice that had responded to therapy versus mice that were non-responders to therapy. And you can see in the volcano plot here, the DIO mice that responded to therapy have increased expression of genes like CD8 alpha, CD3 epsilon, perforin, granzyme. So a very strong effector CD8 T cell response, which is not surprising. And then in terms of genes that were downregulated, we saw things like IL-1 beta, IL-1 receptor, and CCL2, and both IL-1 beta and CCL2 were of interest to us because they are known to recruit tumor infiltrating myeloid lineage cells that can suppress protective CD8 T cell response. So we went back to our mouse model, and we backed up and we looked at an earlier day 15 time point. And here, again, when we took renal tumors from these mice, we can see that they had a non-significant but trending reduction in activated CD8 TILs within those renal tumors from the DIO mice shown here. But they also had a significant increase in myeloid-derived suppressor cells at this time point. And then when we looked within the tumor for concentrations of cytokines and chemokines that are known to be associated with myeloid cell recruitment, we found increases in GM-CSF, IL-1 beta, CXCL1, and CCL2. And we decided to focus on IL-1 beta because it is an early inflammatory signal, and it's known to be upstream of other factors such as CXCL1 and especially CCL2 that can recruit MDSCs. So we asked what would happen if we neutralized IL-1 beta. And I'll take a step back and say before we did that, we also looked at IL-1 beta concentrations as in relation to tumor weight and IL-1 beta concentrations in relation to the frequency of myeloid-derived suppressor cells in these tumors for DIO therapy-treated mice, DIO no therapy-treated mice, and lean therapy and lean no therapy-treated mice. And you can see that we have these nice positive correlations. So as IL-1 beta was increasing, tumor size increased, and the frequency of MDSCs also increased. So then what happened when we neutralized IL-1 beta? So the results are shown over here. So these are all DIO mice. We have a group that received no therapy, a group that received our adenovirus trail plus CPG therapy, and then a group that received the same therapy plus antibodies to IL-1 beta to neutralize it. And what we can see is that the DIO mice that received therapy in this set of experiments, we had only 33% that were defined objectively as responders. But when we added in the IL-1 beta to neutralize that protein, the response rate went up to 58%. And then, not surprisingly, when we administered anti-IL-1 beta, we saw reductions in IL-1 beta concentration specifically within tumor-bearing kidneys. And we also saw reductions in intratumoral myeloid-derived suppressor cells. So in our renal cancer model, we found that obesity decreases protective anti-tumor immunity. And this reflected what we had seen in our renal cancer patients who received anti-PD-1 therapy as standard of care. And then in our mouse model, we further found that this IL-1 beta mediated inflammation seemed to promote therapy resistance. And so, this has really led me to ask, is inflammation the key here? So there was a study published in 2019 from DeGiorgi et al., and they found that low systemic inflammation improved immunotherapy outcomes in renal cancer patients treated with anti-PD-1, regardless of BMI status. And so what I'm showing here, what they showed here were 4 different groups. So group 1 has low inflammation, low systemic inflammation. They did not look at IL-1 beta specifically, but low systemic inflammation and BMI less than 25. Or in group 2, low systemic inflammation, BMI greater than 25. Or individuals who had high inflammation and high BMI, or high inflammation and low BMI. And you can see over here that the individuals who had the best survival were in groups 1 and 2. So low inflammation regardless of their BMI status. So I think this is really important for us to consider, because obesity is a heterogeneous disease state and it's marked by varied comorbidities. So we can't look at one individual who has obesity and assume that physiologically they are equivalent to another individual who has obesity. So we can have individuals who have what I'm going to refer to as simple obesity, so they are metabolically healthy. Or we know that in 30 to 60% of individuals with obesity, they can have elevated systemic inflammation as indicated by high CRP levels. Some individuals with obesity also have hyperglycemia or type 2 diabetes. They can have dyslipidemia, so elevated triglycerides, for example, and they can also have sarcopenia or loss of lean muscle mass. And when we use BMI, I mean, we're guilty of this also, but when we use BMI in these retrospective studies to define obesity, we don't capture this heterogeneity. And so I think we're missing some very important clues about what's happening in these patients. So as I said at the beginning, our initial question was very simple. Does host obesity impair immunotherapy outcomes? And I think now that this is the wrong question to be asking, which brings me back to my optical illusion. So I think if we look at obesity in one way, we might see the lovely young wife, whereas if we look at obesity or define it in a different way, we might see something more akin to the aging mother-in-law. And so I think the more important questions that need to be answered now are, when does obesity impair immunotherapy outcomes? When does it improve immunotherapy outcomes, and why? And I think those are really critical questions, and we are a long way, I think, from being able to answer those right now. So I'll stop there and give my thanks to the past and present members of the Norian Lab, especially Dr. Shannon Boyd, Rachel Orlandella, Justin Gibson, and William Turbot, who did the majority of the work on this project. My collaborators from UAB, Tatiana Marquez Lago, Lakshman Nandagopal, Peng Li, Eddie Yang, and Debbie Delamana in our NanoString facility. And then my colleagues from the University of Iowa, Carver College of Medicine, who helped us to acquire tumor samples from renal cancer patients and helped provide us with patient outcome data. And my financial support from the NCI, American Cancer Institute, and my own institution. And I'll stop there.
And our next speaker is Dr. Katherine Cook from Wake Forest School of Medicine. Dr. Cook received her BS in biochemistry from SUNY at Oswego and her PhD in molecular medicine and translational science from Wake Forest. Dr. Cook was recently promoted to associate professor with tenure in the Department of Surgery at the Wake Forest School of Medicine. Her current research program investigates molecular mechanisms of obesity-mediated breast cancer and dietary interactions influencing therapeutic responsiveness. Current research interests are focused on the impact of the microbiome on modulating inflammation, metabolic syndrome, and breast cancer risk. She has over 45 peer-reviewed publications in the area of breast cancer, and she is currently funded by grants from NIH, the American Cancer Society, Susan G. Komen Foundation, and the Department of Defense. Welcome, Dr. Cook.
Good morning, everyone. My name is Dr. Katherine Cook, and I'm an associate professor in the Department of Surgery at Wake Forest School of Medicine in North Carolina. I'd like to thank the meeting organizers for inviting me to discuss my group's work on the microbiome and how crosstalk between diet and the microbiota influences immune checkpoint therapy responsiveness in breast cancer. So first, a brief introduction on immune checkpoint blockade therapies. Immune checkpoint proteins are attractive targets of significant clinical interest to enhance T-cell anti-tumor function. Activation of inhibitory immune checkpoint protein signaling, such as CTLA4, PD-1, or PD-L1, impairs T cells to activate, recognize, and destroy cancer cells, thereby enabling tumor growth. Several therapies targeting the inhibitory immune checkpoints have been developed and are currently used in the clinic. These include ipilimumab, pembro, nivo, and atezolizumab. While immune checkpoint blockade therapies are more often used in the treatment of melanoma, non-small cell lung carcinoma, and renal cell carcinoma, seminal work establishing immune checkpoint blockade efficacy in triple-negative breast cancer, such as the KEYNOTE-355 and the IMpassion130 clinical trials, should be noted. In KEYNOTE-355, over 800 women with locally recurrent or metastatic triple-negative breast cancer were enrolled. Women were treated with either placebo and chemotherapy or pembro and chemotherapy, and the study reported a significant increase in median progression-free survival in women treated with the ICB and chemo. In IMpassion130, approximately 900 women with unresectable local advanced metastatic triple-negative breast cancer were enrolled. Women were treated with placebo and nab-paclitaxel or PD-L1 and nab-paclitaxel, and the study reported a significant increase in the median progression-free survival in women treated with the PD-L1 blockade and chemotherapy. With these landmark clinical trial outcomes, in 2019, the FDA granted accelerated approval for PD-L1 and chemotherapy for the treatment of locally advanced and metastatic triple-negative breast cancer, making atezolizumab the first immunotherapy approved for breast cancer. In 2020, PD-1 blockade in combination with chemotherapy was also approved for locally advanced and metastatic triple-negative breast cancer. This was followed by the FDA approval of PD-1 therapies in combination with chemotherapy as a neoadjuvant therapy for triple-negative breast cancer in 2021. It's important to note that the current status of immune checkpoint blockade therapy in breast cancer is limited to triple-negative breast cancer. The majority of breast cancer cases, comprising between 60 to 70% of all diagnoses, are estrogen receptor alpha-positive breast cancers. This subtype of breast cancer classically exhibit a non-inflamed or an immune cold phenotype and are less likely to respond to immune checkpoint therapy blockade, as was demonstrated in KEYNOTE-028, which contained a subcohort of ER-positive breast cancer cases that displayed limited efficacy of PD-1 blockade, thereby limiting the use of immune checkpoint blockade therapies to just triple-negative breast cancer subtypes. So of recent interest is the role of the microbiome in facilitating therapy and immune responses. The microbiome is the totality of microorganisms that live on and within the human body. These include bacteria, viruses, fungi, protists, and phages. Of these groups, the most well-studied is the bacterial microbiome. It is estimated that there are approximately 39 trillion bacterial cells living on or within our bodies. with the bulk of these microbes being localized to the gut. The makeup of the average human body comprises about 30 trillion cells, thereby making our body only about 43% human. Therefore, it's unsurprising that these microbes influence the development of disease. Several key points about the bacterial microbiome is: 1, your body is mostly microbes. 2, microbes are influenced by many factors, including diet, drug administration, and genetics. And 3, obesity reduces the gut microbiome diversity, or the total number of different types of bacteria present. While it's a relative recent interest to study the microbiome as a potential immune modulator for the treatment of cancer, it's important to note the historical context. This is actually not a new concept. In the 1890s, Dr. William Coley developed an approach to treat osteosarcoma that involved injecting patients with a mixture of heat-killed streptococcus bacteria, which they were called Coley's toxins. He discovered this approach when he came across a case study of a patient with inoperable sarcoma whose tumor completely regressed after developing a severe bacterial skin infection. It's estimated that Coley himself injected more than 1,000 cancer patients and published 150 papers related on this topic, which arguably gives Dr. Coley the title of father of immunotherapy. Several key clinical studies have investigated the bacterial gut microbiome population as a regulator of ICB response. While highly dependent on geographical location of the study, the cancer type, and the treatment type, studies analyzing the gut microbiome populations of patients prior to therapy initiation and correlating microbes with responsiveness have identified a consortium of immune checkpoint blockade therapy responder-associated microbes. The most commonly identified bacterial populations associated with a favorable ICB response include Akkermansia mucophilia, Bifidobacterium longum, and Faecobacterium parnitzi. The use of fecal microbiome transplant has also recently demonstrated to improve ICB responsiveness. In 2 independent clinical studies, authors showed a fecal microbiome transplant from melanoma patients that responded to PD-1 blockade into melanoma patients that previously displayed PD-1 resistance prior to their retreatment with PD-1 antibody, indicates that fecal microbiota transplants, or FMTs, enhance immune checkpoint blockade therapy responsiveness. These data demonstrate the efficacy of shifting the gut microbiome in humans to improve ICB therapeutic efficacy. While the bulk of the studies investigating microbiota influences on ICB responsiveness have been in melanoma, recently we've collaborated with Dr. Lyse Norian to characterize the impact of obesity on PD-1 responsiveness in breast cancer. They showed that PD-1 blockade in obese animals revitalizes the immune compartment to improve anti-tumor immune checkpoint blockade responses in a preclinical syngeneic triple-negative breast cancer model. This was associated with an elevated M1-like macrophage population and increased CD8 positive T cells. They characterized the gut microbiome in anti-PD-1 treated animals and showed that this was associated with increased proportional abundance of Akkermansia, Bifidobacterium, and Lactobacillus, among others. These data also highlight that the previously identified ICB responder consortium of microbes are also similarly correlated with PD-1 responsiveness in the preclinical breast cancer models. When planning preclinical immunotherapy and microbiome studies, an important consideration is the model. Using syngeneic models enables an intact immune system response. Also, having multiple syngeneic models derived from different mouse strains will greatly enhance study strength, as the bulb C and the C57 Black 6 mouse strains have different immune bias. In our studies, we use the EMT6, which is a triple-negative breast cancer cell line in the bulb C background, and the EO771, which is also a triple-negative breast cancer model in the C57 Black 6 background. Both are mirroring triple-negative breast cancer models that display enhanced immunogenicity and respond to immune checkpoint therapy blockade. We also use 3 different diets in our studies. We use the standard burden control diet, which is 10% kilocalories from fat. We also have designed a Western and a Mediterranean-like diet representing human dietary patterns. Both the Mediterranean and the Western diet are 45% kilocalories from fat. However, the source of the fat differs between the diets, with Mediterranean diet fat sourced from olive oil and fish oil, and the Western diet fat sourced from anhydrous milk fat and corn oil. Western diet has elevated saturated fat content, a high N6 to N3 PUFA content, increased sucrose, and a higher sodium levels when compared to the Mediterranean diet. We performed echo MRI and glucose tolerance tests in both the C57 Black 6 and the bulb C mice that were consuming these diets to demonstrate Western and Mediterranean diet effects on metabolic parameters. Both diets result in increased body fat composition compared with control diet. However, only Western diet consumption led to impairment of glucose homeostasis, as indicated by the glucose tolerance test. To determine whether diet modulated PDL response, we placed 7-week-old bulb C mice on a control Mediterranean or Western diet for 4 weeks to establish diet influence shifts on the metabolic and gut microbiome parameters. We then injected EMT6 triple negative breast cancer cells into the mammary fat pad of these animals. 1 week later, mice on each diet were treated with IgG control antibody or an antibody to block PDL1. Tumor volumes were recorded every 3 days for 21 days. There was no significant difference in the tumor volumes in the IgG control treated mice consuming either a control or Western diet. However, mice consuming a Mediterranean diet did display reduced tumor volume and tumor weight. While PDL1 treatment significantly reduced tumor volume compared with IgG treatments within all diets, consumption of a Western or Mediterranean diet significantly enhanced immune checkpoint blockade therapy response compared to the control diet. These trends were also observed with the tumor weight at the study endpoint, where Western and Mediterranean diet-fed mice treated with PDL1 antibody displayed significantly reduced tumor weight compared with the control diet-fed PDL1 treated mice. These diets demonstrate that diet modulates immune checkpoint therapy efficacy in the EMT6 triple negative breast cancer model. We also performed a second model using the E0771 luciferase triple negative breast cancer model in the C57 Black 6 mice. Similar to the last model, we placed 7-week-old female C57 Black 6 mice on a control Western or Mediterranean diet for 4 weeks before injecting the E0771 luciferase triple negative breast cancer cells into the mammary fat pad. 9 days later, these mice were treated with control IgG or PDL1 antibody, and tumor volume was measured every 3 to 4 days for 2 weeks. As you can see, the diet had no effect on the control IgG tumor growth. Mice consuming a control or Mediterranean diet displayed a similar anti-tumor response to anti-PDL1 treatment. Mice consuming a Western diet displayed enhanced anti-tumor responsiveness to PDL1 treatment when compared with the other 2 diets. This was also observed in tumor wet weight. These data demonstrate that Western diet potentiates PDL1 responsiveness in the E0771 triple negative breast cancers model. We next performed metagenomic sequencing on DNA isolated from fecal matter collected in the EMT6 study before treatment at day 0, and then at the end of this treatment at day 21. The principal coordinate analysis indicates samples within dietary patterns grouped together. Moreover, significant differences in the Western PDL1 and the Mediterranean PDL1 pre and post-treatment, but not in the control diet or in the IgG treatments, indicate therapeutic shifts mediated by immune checkpoint blockade therapy in the higher fat content diet-consuming mice. Taken together, these data indicate that diet is the main regulator of the gut microbiome, and PDL1 treatment within each diet shifts the gut microbe populations as well. At the species level, diet regulated several microbes. Control diet-fed mice displayed higher bacterial beta-telmicrons, Lactobacillus jasoni, and Clostridium. Mediterranean diet-fed mice displayed elevated Lactococcus lactis. Moreover, Western and Mediterranean diet-fed mice displayed approximately 25 to 45% proportional abundance of Akkermansia immunocephalia, as highlighted in the red box, which is one of the immune checkpoint blockade therapy responder-associated microbes. suggesting that the potential enhancement of the anti-tumor PD-L1 activity in the Western and Mediterranean diet-fed EMT6-bearing mice may be due to the higher abundance of this bug. Administration of PD-L1 antibody resulted in a shift of proportional abundance of certain bacterial populations at the genus level within each diet. Treatment with immune checkpoint blockade therapy in control diet-fed mice increased Eldocrucia, included Clostridium abundance. Within the Mediterranean diet-fed subjects, treatment with PD-L1 antibody increased Anotronchus, Eubacterium, and Roseoburia populations. Further studies are needed to explore the effects of the anti-PD-L1 treated regulated microbes on inflammation and efficacy. The gut microbiome is more than just bacteria. While we did not observe any significant modifications to the viral, fungal, or protist populations, we did notice a notable change in the bacteriophage populations. Most striking was the difference in the alpha diversity between the low-fat control diet and the higher fat content containing diets. Animals fed a Western or Mediterranean diet expressed a greater number of different types of detectable phages than the control diet-fed animals. While diet is the main determinant of the phage populations, PD-L1 antibody administration significantly shifted the phagome within the Western and Mediterranean diet-fed animals. Within the Western diet-fed animals, immune checkpoint blockade therapy increased the temperate lactococcus phage BIL-310 and decreased the temperate lytic lactococcus phage BIL-285. In the Mediterranean diet-fed animals, PD-L1 antibody treatment increased the temperate lactococcus phage BIL-311. Indeed, within the Western diet-fed animals, gut lactococcus lactis abundance negatively correlates with the temperate lytic phage BIL-285 abundance, suggesting the potential phage-mediated regulation of the lactococcus lactis population by immune checkpoint therapy. To focus on potential gut microbe regulation of therapeutic response, we performed a fecal microbiome transplant. 8-week-old female bulbsy mice were fed a standard control diet and placed in antibiotics, which was a mixture of streptomycin, colistin, and ampicillin mixture for 3 days. We have previously shown that this combination of antibiotics ablates the gut bacterial microbiome population. We then restored the gut microbiome by giving mice either a control diet-derived fecal microbiota transplant, a Western diet-derived fecal microbiota transplant, or a Mediterranean diet-derived fecal microbiota transplant by oral gavage. FMTs were prepared fresh feces collected from non-tumor-bearing bulbsy mice on each diet. We've previously demonstrated diet-derived fecal microbiota transplant efficacy at shifting the gut microbiome populations in our recent cancer research publication. EMT6 cells were then injected into the mammary fat pads, and 1 week later, mice on each FMT were given a control IgG or PD-L1 antibody treatment, and the tumor volume was measured every 3 to 4 days. Diet-derived FMT had no effect on the primary EMT6 tumor growth in the control IgG-treated animals. Mice that received Akkermansia immunosyphilic enriched FMTs from Western or Mediterranean diet-fed mice displayed increased anti-tumor PD-L1 response when compared to the control diet-derived FMT PD-L1-treated animals. Taken together, these data suggest that the increasing gut Akkermansia populations may enhance immune checkpoint therapy efficacy in triple-negative breast cancer. So I briefly want to shift gears and address the concept of a tissue-specific microbiome. Outside of the gut, other organs have been shown to contain their own bacterial microbiomes. The breast was shown to contain bacteria in 2014. Moreover, studies went on to show that the breast cancer can shift the breast tissue microbiome. Urbanic et al. Showed increased staphylococci in breast tissue from women with breast cancer when compared with healthy controls. Another group demonstrated reduced lactobacillus in breast tissue from women with malignant breast cancer. However, these initial studies did not control for BMI. Since diet is the main determinant modulating the gut microbiome, we asked the question whether diet could shift the breast microbiome. To answer this question, we partnered up with Dr. Carol Shively, who had previously performed a dietary non-human primate model where female macaques were fed a Western or Mediterranean diet modeled to represent human dietary patterns. These primates were fed their diets for 2.5 years. We showed that diet modified both the gut microbiome, which we previously published in Microbiome of this year, and the breast microbiome previously published in some reports. Since these studies were previously published, I won't go into depth. However, it's important to note that habitual consumption of a Mediterranean diet led to a 10-fold increase in the breast probiotic lactobacillus abundance, thereby demonstrating that diet can regulate the bacterial microbiome populations in gut distal organ sites like the breast. As a follow-up to this study, we wanted to determine whether body adiposity could be influencing breast health. To do this, we went back to our non-human primate cohort and subdivided the dietary cohorts based upon body fat composition, as our non-human primates were given DEXA scans at the end of the study. We then divided the subjects into tertiles based upon body fat composition to form the Mediterranean lean, with an average of about 8% body fat, the Mediterranean heavy, which had an average of about 23% body fat, a Western lean, which has an average of 12% body fat, and a Western heavy with an average of about 39% body fat groups. We then performed untargeted metabolomics on SNAP frozen breast samples from the heavy and lean diet-fed subjects. We identified over 246 metabolites regulated by diet and 189 by body weight diet interaction. However, only 3 metabolites were similarly regulated by body fat composition, regardless of dietary pattern consumption. Vitamin A, or retinol, is very lipophilic. Therefore, the increased concentrations observed in the heavy non-human primate breast tissues may be just a result of the increased breast adipocyte size. However, a recent publication by Wu et al. In 2021 demonstrated commensal probiotic bacteria in the gut can process dietary vitamin A into retinoic acid, thereby potentially linking bacterial-mediated metabolism to retinol bioavailability. The increased lactate and maltose observed in the lean non-human primate breast tissue could be a result of microbial metabolism as well. Certain bacteria produce lactate as a metabolic endpoint, and other bacteria populations contain amylase, which is an enzyme that can metabolize starch to form 2-glucose and 1-maltose. We then went back to our non-human primate mammary gland 16S sequencing results and reanalyzed the microbiome by diet and adiposity. We noticed regulation of lactate-producing bacteria by adiposity in both dietary patterns, and particularly lean non-human primates, regardless of diet, displayed elevated abundance of lactobacillus within their breast tissue. Although it's important to note that the proportional abundance of lactobacillus is an order of magnitude higher in the Mediterranean diet-fed lean subjects when compared with the breast tissue from Western diet-fed subjects. Lactate is known to be immunosuppressive. Therefore, the decreased lactate and lactate-producing bacteria present in breast tissue from the obese, heavy, non-human primates may actually represent a more immune-favorable microenvironment that could enhance ICB response. To address whether obesity regulates the human breast microbiome, we collaborated with Dr. Neil Iyengar to perform a pilot study characterizing the breast microbiota populations in 20 women with known BMI. DNA was isolated from frozen breast tissue and 16S sequencing was performed. Samples were categorized into obese with a mean BMI of 34.7 or lean with a mean BMI of 22. Trends in populations at the family level indicate elevated enterobacteriaceae and lactospiraceae and breast tissue from obese women, while elevated corneobacterium and staphylococcus abundance in microbes were identified in lean women's breast tissue. At the species levels, an obese women's breast tissue displayed elevated amounts of several immune checkpoint blockade responder-associated microbes, including Akkermansia and Bifidobacterium, thereby raising an interesting question of whether tissue-specific microbiomes may influence immune checkpoint blockade therapy responsiveness. So in summary, Western or Mediterranean diet consumption enhanced PD-L1 blockade therapy efficacy in the EMT6 model of triple-negative breast cancer. Western diet consumption enhanced PD-L1 blockade therapy responsive in the E0771 model of triple-negative breast cancer. Western and Mediterranean diet consumption is associated with higher levels of gut Akkermansia and diet and immunotherapy shift the gut bacteriophage populations. In Western or Mediterranean diet-derived fecal transplants potentiates PD-L1 blockade efficacy when compared with controlled diet-derived fecal transplants in the EMT6 model. Lastly, obesity increases the proportional abundance of ICB response-associated microbes, Bifidobacterium and Akkermansia, within the breast tissue of patients. So with that, I would first like to thank my lab manager and animal model expert, Mr. Wilson, and a very talented graduate student, Ms. Clear, for their work on this project. I also would like to thank our internal and external collaborators, as science is very much a team activity. Lastly, the funding agencies and foundations I would like to thank that currently fund my group for making all this research possible. Thank you so much.
And our last speaker, but certainly not least, is Dr. Richard Simpson from the University of Arizona. He is professor in the Department of Nutritional Sciences and holds joint appointments in pediatrics, immunobiology, the Arizona Cancer Center, and the Bio5 Institute. Dr. Simpson is known internationally for his work on exercise and the immune system. Specifically, his research interests are concerned with the effects of aging, stress, and exercise on the immune system and the role of the adrenergic receptor signaling on immune cell redistribution and activation. His current research is supported by NASA, the National Cancer Institute, and industry. He is currently president-elect of the International Society of Exercise Immunology, and he would want me to tell you that the next meeting is in person in Arizona in 2022. And he is a fellow of the American College of Sports Medicine. Dr. Simpson.
Okay, thank you for the introduction, Katie. As was mentioned, my name is Ricky Simpson. I'm a professor at the University of Arizona. And what I'm really interested in speaking to you about today is how exercise could be used as an adjuvant for immune cell therapies. And we're really trying to explore the mechanisms by which exercise can serve as this adjuvant. And by that, we're really looking at beta adrenergic receptor signaling. So that's what I'm going to talk to you about today. Okay, so we know exercise training can reduce cancer risk and also improve outcomes in many cancer survivors. But we're really just starting to get an idea of what those underpinning mechanisms actually are. So if you look at the table on the right-hand side, this is a review paper that was published by a group in Copenhagen in Cell Metabolism a few years ago. And essentially, they were just summarizing all the mouse models that have been used to assess the impact of exercise training on tumor growth. And you can see here that many different tumor models have been used, both in immunocompetent mice and also looking at human tumors in immunoincompetent mice. And we can see that exercise training is reducing tumor growth anywhere between 30 and 70%. And that's been quite consistent across many different studies. Now, in terms of the mechanisms that are involved, tumor perfusion has been proposed, as has changes in vascularity and angiogenesis, hypoxia, changes in metabolism and inflammation and hormones. And these are all being studied at the moment by various groups. But what we're interested in really is the effects of exercise on the immune system and how the immune system can be manipulated by exercise to improve anti-tumor immunity. And in what we know with exercise is that exercise causes a release of catecholamines, which may have either direct or indirect effects on tumor growth. And then also when we exercise, our muscle releases certain cytokines that have been termed myokines. And then these myokines are also involved in the immune actions against the tumor. So one of the messages I would like to convey in this seminar is that every single exercise bout you do matters, because we have these very transient spikes in hormones and immune responses that over time accumulate into the chronic effects of long-term exercise training. So to give you an example, every time we exercise, our muscle releases cytokines, namely interleukin-6, interleukin-7, and interleukin-15. And they do spike and come back to normal levels very quickly after exercise. We also see increases in catecholamines like epinephrine and norepinephrine. And it's these spikes in exercise factors that we see with each bout of exercise that can contribute to reduced tumor growth and reduced cancer risk. Now, whenever myokines and catecholamines are released, we also see an almost instantaneous mobilization of immune cells. And this is a very robust, a very massive mobilization. And all immune cells in circulation will increase in number, but in particular, we see pretty large increases in the number of natural killer cells, followed by T cells, and then lastly, B cells. But interestingly, as soon as we stop exercising, these cells rapidly leave the blood compartment. And this can happen within minutes of stopping exercise. And it's at this point the myokines are very important, because the myokines are actually telling our immune cells that have been mobilized to traffic around our tissues to perform these immune surveillance roles. And in the context of cancer, we see that these immune cells actually traffic towards the tumor and participate in inhibiting tumor growth. So this is a paper that was published maybe 5 years ago now, but it's a really good example of how this mobilization and redistribution of immune cells with every single bout of exercise is important. And this is a mouse model. It's a B16 melanoma tumor model. So it is a skin cancer tumor, but it does metastasize to the lungs. And then what we can see here is in mice that were given this tumor, they were randomized to a non-exercise group, which is the control, or a group that were given access to a running wheel in their cage. And then we can see that the mice that had access to the running wheel had substantially less tumor volume compared to the controls. And they also had fewer tumors in the lungs compared to the controls. But interestingly, when they looked inside the tumor, they found that the exercise group had more than twice as many natural killer cells. So these are potent anti-tumor cells that our immune system is armed with, compared to the mice that were in the control group. So we've known for quite some time that exercise mobilizes these immune cells via adrenergic mechanisms. And we also know that if we give beta blockers to humans, we can actually abrogate this mobilization of immune cells with exercise. So what this group decided to do is they decided to give the exercise in mice propranolol, which is a non-selective beta agonist. And we can see here that the inhibitory effects of exercise are completely abrogated when the mice receive this non-selective beta blocker. And then to look at the other side of the coin, instead of exercising the mice, they just decided to give them daily shots of epinephrine. And when they gave the mice daily shots of epinephrine, they also saw a reduction in tumor volume that was akin to exercise. And then if they looked at the natural killer cells in the tumor, then it tended to mirror what they were seeing with both the beta blocker and the beta agonist. So we also know that skeletal muscle regulates immunity. So I mentioned that these myokines that were released during exercise, like IL-6, IL-7, and IL-15, they're known to have very important effects on many aspects of the immune system. So they can help maintain monocyte and macrophage function. We know that cytokines such as IL-15 can increase the expansion of natural killer cells and make them more cytotoxic, which essentially means they'll be better at recognizing and killing tumor cells. But cytokines like IL-7 as well as IL-15 are also very important for maintaining the peripheral T cell repertoire. So essentially, we can maintain our naive T cell compartment with these cytokines that are released with exercise. And then we can also produce new T cells from the thymus as a result of the IL-7 that is elevated with exercise training. And then quickly going back to the study I showed you previously, the Pedersen group were interested to know if the IL-6 that they were seeing increase with exercise was playing a role in suppressing tumor growth with exercise. And then we can see here that in the mice that were exercising versus the controls, there was an inhibition of tumor growth. But when they blocked IL-6 in vivo, we can see that the exercise effects were abrogated. And this also prevented the influx of natural killer cells. So what they really concluded from this study is that adrenergic receptor signaling mechanisms are important for mobilizing immune cells into the periphery, but it's the myokines that are released from muscle during exercise that play an important role in helping these cells traffic towards the tumor. So we've actually shown this adrenergic effect in humans. So these were healthy individuals that we asked to perform 30 minutes of cycling exercise at 10% above their lactate threshold. And what we did here is we asked them to consume either a placebo or a selective beta-1 antagonist. So we gave them bisoprolol, or we gave them a non-selective beta-1 and beta-2 antagonist in the form of nadolol. And they ingested these drugs or placebo 3 hours before performing the 30-minute bout of exercise. And then we collected blood before they got the drug, after they got the drug, before they started exercise, immediately at the end of exercise, and then an hour into recovery. And I should point out that this was a killer cells, and these effector memory CD8 T cells was blocked when we gave the individuals the non-selective beta antagonist. So this was nadolol. But interestingly, when we blocked the beta-1 receptor, it actually increased the mobilization. So we can see that we actually get more natural killer cells in the blood if we block the beta-1 adrenergic receptor, but not the beta-2. And what we really think is going on here is that whenever we block the beta-1 receptor, it's actually making more of the epinephrine available for beta-2 signaling. So it's actually augmenting the mobilization. And we think that this is very important for the types of beta blockers that cancer patients are being prescribed. And I'll talk a little bit about that later. So we know that these immune cells that are mobilized with exercise have very potent anti-tumor properties compared to their counterparts in resting blood. So questions that we've been asking over the last 5 years or so is can a single exercise bout actually augment cancer cell therapies? And one of the cell therapies that we're really interested in is allogeneic cell therapies, such as donor lymphocyte infusions. So if you don't know, donor lymphocyte infusions tend to be given to recipients of hematopoietic stem cell transplantation after they receive their stem cell therapy. And the donor lymphocyte infusions, or DLIs, are used to either prevent or treat relapse. So for the most part, these are patients that have leukemia. But we can see here with this diagram is that the lymphocytes are typically collected from a donor under resting conditions, and then they're collected by an apheresis machine and then subsequently transferred to the cancer patient. But because we know that just a single bout of exercise mobilizes very potent anti-tumor immune cells into the blood, we've been asking the question, well, instead of collecting these immune cells from the blood under resting conditions, can we actually collect them from the donor when they're exercising? And then because of the increase in epinephrine and mobilization of blood lymphocytes, we potentially could have a better therapeutic product to give to cancer patients. So I won't go through all of these in detail, but this is a very detailed 8-color flow cytometry panel that we did in our lab. We've looked at changes in a whole range of monocytes and T cell populations with exercise, and then we've looked at a whole range of natural killer cell subtypes in response to exercise. And what you can see here is that pretty much every single immune cell subtype increases numerically in blood, but the magnitude of the mobilization varies depending on the immune cell population. But what we've really concluded from this work is that exercise preferentially mobilizes both natural killer cells and CD8 positive T cells with an effector phenotype. And what this really means is that they have an enhanced ability to recognize and kill various tumor cells. And then they also have an enhanced ability to respond to chemokines. So that means their ability to traffic through tissues is improved. We also know that they respond more readily to cytokines, and they have an enhanced ability to proliferate and persist. So overall, this phenotypic signature would indicate that these mobilized cells are going to be much better therapeutic products for adoptive cell therapies. So we also know that just exercise in individuals can massively change the composition of lymphocytes in peripheral blood. And by changing this composition, it would actually be more advantageous to recipients of allogeneic cell therapies. So there are really two components of an allogeneic cell product. There's the graft versus leukemia effect, which is something that you want. So you want to have more of those cells that can recognize and kill leukemic cells. But on the flip side, you also have a side effect called graft versus host disease. And this is when the immune cells from the donor start to attack healthy tissue in the cancer patient recipient. So all in all, what we want to do is try and reduce those cells that cause graft versus host disease and increase the cells that cause graft versus leukemia. So I don't have time to go through all of these different subtypes in details, but some of the cell types we know cause graft versus host disease is CD4 alpha beta T cells and B cells, whereas the cells that more preferentially cause graft versus leukemia effects are the natural killer cells and memory populations of CD8 T cells. And if we just, quickly group these into favorable cells and less favorable cells, then we can see in resting blood, the less favorable cells make up about 50% of all the lymphocytes in blood. Whereas with exercise, we see that this is substantially reduced. Okay, this gets down to about 24%, and conversely, the favorable cells go up. So just by shifting the composition of these cells, we think will be advantageous for cancer patients. So we decided to test this using a humanized mouse model. So we used the NOD-SCID mouse, which has a human IL-15 knock-in. So this mouse model has no immune system of its own, which means we can quite simply take human cells, transplant them to these mice, and then the human cells will grow and persist in the mice. The reason we use the human IL-15 knock-in is because some immune cells, like natural killer cells, for example, don't last very long if they don't have that cytokine to feed them. So the fact that this mouse makes endogenous human IL-15 allows the natural killer cells and other immune cells, such as gamma delta T cells, to persist in the mouse after we adoptively transfer the human cells. And what we did here is we irradiated the mice, and then a day later we injected the mice with peripheral blood mononuclear cells that we collected from individuals under resting or exercise conditions. So the exercise they did was just 30 minutes of cycling at 10% above lactate threshold. And then we injected these PBMCs into the mice, and then a day later we challenged them with the K562 chronic myeloid leukemia cell line. And this cell line was luciferase tagged, which meant we could monitor growth over 40 days by bioluminescent imaging. And then what we found is that when we injected PBMCs that we collected after exercise compared to rest, we actually saw an increased overall survival in the mice that received the exercise mobilized lymphocytes. So you may look at this and think, well, okay, the mice that got tumor on their own actually live longer, and that's actually true. And that's really because of that side effect I mentioned, that graft versus host disease, because when we give human immune cells to a mouse, then the human immune cells are going to recognize the mouse tissue's form and attack it. So it is one of the limitations of the model. But when we just compare the resting to the exercise mobilized cells, then we do get a survival advantage. But when we look at tumor burden, so in the mice that received the K562 tumor alone, we can see that the tumor grows quite readily. And then just adoptively transferring the human PBMCs inhibits tumor growth. But you can see the overall inhibition was greater when the mice got the exercise mobilized PMCs. And then if we look at the tumor-free survival curve here, we can see that at the end of the experiment, so after 40 days, about half of the mice that were still alive and received exercise mobilized lymphocytes, half of them were tumor-free, whereas all the mice that received resting lymphocytes that were alive at the end of the experiment, all of those mice had discernible tumor growth. We also looked at the effects of the PBMCs in non-tumor bearing mice just to see if there was any differences in human cell engraftment or xenograft GVHD, and we actually found no difference. And then we're currently doing single cell RNA sequencing to identify some of the underlying mechanisms that could be underpinning this. So in addition to donor lymphocyte infusions, which are relatively rudimentary, so you've probably heard of new therapies now, such as CAR T cells and cytokine-induced killer cells and various other cell products that cancer patients are receiving. And then one of these cell types is gamma delta T cell therapy. And this is a therapy that's more popular in Europe. But the reason gamma delta T cells are attractive is because they have potent anti-tumor properties and they don't cause graft versus host disease. So essentially you can take gamma delta T cells from one person and transplant them to a completely different patient. And then the risk of graft versus host disease is almost zero. But with gamma delta T cells, they are a very rare population. They only make up about 5% of all the T cells in blood. And as I mentioned, they function across MHC barriers, so they don't cause GVHD. But their function is regulated by an array of activating and inhibitory receptors in a manner that is akin to natural killer cells. They're very easy to grow. We can take them from the bloodstream of a cancer patient or a donor, and we can put them in a cell culture flask, and we stimulate them with aminobiphosphonates such as zoledronic acid and some growth cytokines such as IL-2. And then after about 14 days, we end up with massive numbers of these gamma delta T cells that we can adoptively transfer either back into the patient if we're using their own cells, or into a completely different patient if we've collected them from a donor. We can also give zoledronic acid and IL-2 in vivo, so we can just inject zoledronic acid in particular into the patient, and then this causes the tumor to release IPP, which activates and expands gamma delta T cells and actually sensitizes the tumor to the gamma delta T cells, so that they become more cytotoxic. So these are procedures that are currently being used in the clinic, but we wanted to know if exercise could improve the ex vivo expansion of these gamma delta T cells, and of course their efficacy both in vitro and in vivo. So what we decided to do here is ask the question whether or not exercise and beta-2 adrenergic receptor activation can augment the ex vivo expansion and function of gamma delta T cells. So we had 3 cohorts that participated in this study, and I should mention that this study is still ongoing, and we're a little bit further along with some of the analyses than we are with others. But we did have an exercise cohort who simply exercised for 30 minutes at 10 or 15% above their blood lactate threshold. And then we collected blood before, during, and 1 hour after exercise. We also had a cohort of participants who completed exercise with and without beta adrenergic receptor blockade. So this is the exact same design I explained earlier. It was a crossover design. It was randomized, double-blind, placebo-controlled, where the individuals consumed either a beta-1 selective antagonist or a non-selective antagonist prior to exercise. And then we collected blood at the exact same time points. Now, because we believe that the mechanisms of exercise are all adrenergic receptor mediated, we wanted to use an experimental model where we could activate adrenergic receptors without exercise. So what we did here is we injected healthy individuals with the non-selective beta agonist isoproterenol. So this is a drug that's been around for a long time. It's actually used to treat heart failure. And as you would imagine, it evokes a physiological response that's similar to exercise. So we see increases in systolic blood pressure. We also see increases in heart rate. But you can see here, I was actually a subject from my own research here. I'm being injected with isoproterenol, and I'm clearly not exercising. But we wanted to know if that adrenergic stimulation would give us a similar response. So we collected gamma delta T cells from the blood. So I should mention that this is the exercise cohort. And as you can see, we get more gamma delta T cells in the blood when the individuals exercise. But when we take these gamma delta T cells out of blood and expand them in culture over 14 days, we can see that we get a much larger expansion when we collect the gamma delta T cells from blood after exercise. In terms of the composition and so forth, I won't go into this in too much detail, but we didn't find any difference between the rest and exercise expanded gamma delta T cells, which is actually a good thing. But when we put the gamma delta T cells in a cell culture flask with cancer cells, and this was the K562 myeloid leukemia cell line, we did find that the exercise mobilized expanded gamma delta T cells were much better at killing K562 tumor cells compared to the gamma delta T cells expanded under resting conditions. And then when we added zoledronic acid to the cell cultures, this is a way to sensitize the tumor to the gamma delta T cells. You can see that the overall effect was increased killing, but the magnitude of the response was greater for the gamma delta T cells we expanded after exercise. So overall, this told us that if we collect gamma delta T cells from blood after exercise, we can expand more of them. And then these cells that we expand are much better at recognizing and killing tumor target cells. In the beta blocker cohort, we found, as expected, that when we give nadolol, so this is the drug that will block the beta-2 receptor, but not bisoprolol, which leaves the beta-2 receptor intact, we can see that the mobilization of gamma delta T cells was abrogated with nadolol. And then, of course, the expansion of the gamma delta T cells was abrogated with nadolol. So this told us that the effect was most likely beta-2 adrenergic dependent. So when we took the gamma delta T cells and transplanted them into tumor-bearing mice, so again, we used the exact same model that we did with the donor lymphocyte infusions, just this time we gave purified expanded gamma delta T cells instead of mixed lymphocytes. And then what we found is that the gamma delta T cells that we expanded after exercise did a slightly better job than gamma delta T cells expanded under resting conditions. But when we injected the mice with zoledronic acid, which mimics the patient's situation, we found that the tumor growth was a lot less in the mice that received exercise-expanded gamma delta T cells in combination with systemic zoledronic acid therapy. And we can see this more clearly here in the tumor-free survival curve, where the mice that get the exercise-expanded gamma delta T cells in combination with zoledronic acid had better survival than mice that received zoledronic acid plus the gamma delta T cells expanded under resting condition. So moving on to the isoproterenol infusion model, again, we just collected the gamma delta T cells before and after isoproterenol infusion. We expanded the gamma delta T cells over 14 days. And then when we co-cultured them with K562 cells, we can see that the isoproterenol mobilized cells that we expanded did a much better job killing the K562 cells. Again, this is exactly what we found with exercise, so we're able to replicate that. And then when we transplanted the gamma delta T cells to the mice that were bearing the K562 leukemia, we can see that given mice zoledronic acid on its own doesn't have any impact on tumor growth if there's no gamma delta T cells present. But we can see that the isoproterenol expanded gamma delta T cells were much better at controlling tumor growth than the gamma delta T cells that we expanded under resting condition. And then, of course, when we injected mice with zoledronic acid, then we saw the best effect. So here we can see that mice that received isoproterenol expanded gamma delta T cells with zoledronic acid had almost a complete abrogation of tumor growth. And the isoproterenol effects were actually were more impressive than the exercise effects. So we wanted to try and understand what the mechanisms are at play. So the first thing we did is we compared the resting and exercise mobilized gamma delta T cells to see if there were any phenotypic shifts that could be explaining the effect. And what we found is that there was an upregulation of some of these activating receptors, in particular DNAM1, TRAIL, and NKG2D. Okay, these are all receptors that if they're overexpressed, then they're more likely to be able to kill the tumor target cell. And conversely, some of the inhibitory receptors that if they are overexpressed, then the tumor is more likely to evade the immune cell. But these were actually downregulated. So NKG2A, for example, is an inhibitory receptor that was downregulated. We also did some bulk RNA sequencing. We took the expanded gamma delta T cells before and after both exercise and isoproterenol infusion. We sent these to our genomics core and our bioinformatics core. And they did some gene set enrichment analysis, annotated to genomic ontology and KEGG, which are publicly available databases to try and find if there's any particular pathways or gene sets associated with pathways that are enriched due to the treatment. And what we really found is that in the exercise expanded cells, as well as the isoproterenol expanded cells, is we saw upregulations of gene sets associated with oxidative phosphorylation and aerobic metabolism. Cytokine receptor interactions were upregulated. Lymphocyte or NK cell mediated cytotoxicity was upregulated, as was gene sets associated with migration and chemotaxis, and importantly, immune responses to tumor. Whereas we also saw downregulations in markers of T cell differentiation and apoptosis and inflammatory activity, which informed us that the cells that we are expanding after exercise or isoproterenol infusion are more likely to persist after transfer. Okay, so the last set of experiments I want to show you that we did is, I mentioned earlier, we saw upregulations of key activating receptors like DNAM1. So we wanted to know if blocking DNAM1 would actually take away the increased cytotoxic effects that we were seeing with exercise. So what we have here is we have a range of well-known receptors that are expressed by gamma delta T cells. And they're known ligands that are expressed by various tumor cells, including the K562 tumor that we used in this model. So the first thing we did is we blocked DNAM1, and we measured if the exercise expanded gamma delta T cells could still kill the K562 tumor cell in vitro. And then what we found is that by blocking DNAM1, we completely took away the effects of exercise. So the exercise expanded cells were no longer able to kill the K562 cells. So you can see here there's no difference now between the exercise and the resting condition. We also looked at trail. It was another activating receptor we saw regulated, and blocking trail also abrogated the effects of exercise. And then to look at the other side, we thought, well, okay, if we're blocking DNAM1, which is expressed by the gamma delta T cells, the known ligands for DNAM1 are PVR and Nectin-2. They're expressed by K562 cells. So if we block those on the K562, then we should also abrogate the effects. And when we did that, we found that it did. Blocking PVR took away the exercise effects, and also blocking Nectin-2, which is another ligand for DNAM1, took away the exercise effects. So just to summarize what I've covered today then is you know, I hope I've convinced you that exercise can reduce cancer risk and treatment-related complications. More so from work that's previously been published in the literature. But one of the messages I would like to get across today is that every exercise bout matters, and the activation of these biological stress pathways, particularly the pathways that involve beta-2 adrenergic receptor signaling, appear to play a pivotal role. They play an important role because there is a huge mobilization of and redistribution of effector lymphocytes that are known to have potent anti-tumor activities, in particular the natural killer cells and the CD8 positive T cells, but then also the myokines that are skeletal muscle release with every bout of exercise, such as IL-6, IL-7, and IL-15. They appear to play a prominent role in regulating immunity as well, not just by maintaining the peripheral lymphocyte compartment, but also helping those mobilized cells with exercise traffic to and infiltrate tumors. So one thing that we're really interested in our lab is whether or not exercise can have adjuvant effects for cancer therapy. We think that it definitely can. I focus my talk mostly on adoptive cell therapies, but I know there's other groups that are interested in how exercise can augment the efficacy of immune checkpoint inhibitors. There's also groups looking at how exercise can improve the efficacy of chemotherapy. I know there's groups looking at immune constitution after hematopoietic stem cell transplantation, for example. I know there's groups interested in how exercise can serve as an adjuvant for tyrosine kinase inhibitors and various other therapeutics. But we think that adoptive cell therapies are quite important, not just exercising the cancer patients, but also exercising the donors, just because collecting these immune cells from the blood during exercise appears to have advantages for the therapeutic delivery of some of these cell products. But the key question that we're trying to address moving forward is, what are the underlying beta-2 adrenergic receptor signaling pathways? And we're currently using single-cell RNA sequencing to get at that question. Because, as I'm sure you'll appreciate, we can activate the beta-2 adrenergic receptor, but there's so many other downstream signaling pathways at play. And we really need to understand that network in more detail so that we can design better therapeutics to activate these pathways for giving to cancer patients. But a question I like to pose at the end of these talks is, given what we know about how activating the beta-2 adrenergic receptor plays in mobilizing immune cells with exercise and how it can help immune cells traffic to tumors, is how will prescription beta blockers affect the anti-cancer immune effects of exercise? So as I'm sure we appreciate, many cancer patients also have underlying issues that they might be receiving beta blockers for. And if they are receiving non-selective beta blockers like propranolol or carvedilol or nadolol, then potentially they could be putting the brakes on the exercise effect, and they might not be getting the best effects of exercise that they could be in terms of reducing their cancer risk or the risk of relapse, and improving the immune surveillance effects of exercise. So just finally, I just want to acknowledge some individuals. So all of these people here are either past or present members of my lab. A few individuals I'd like to shout out are, first of all, Douglas Diak and Helena Batatina. So they were instrumental in establishing the humanized mouse models in my lab. And then Forrest Baker was a key player in the gamma delta T cell work, actually formed a large part of his PhD dissertation and his postdoc. And then just a quick shout out to the people who pay the bills. Most of my research is funded by NASA and the National Cancer Institute, and also the American College of Sports Medicine. And with that, I'll thank you for listening.
Okay, that was fantastic. Really, really wonderful presentations. Thank you to all of our speakers. And thank you to everyone who has been submitting questions. We actually have quite a few questions here, and I'm thrilled to be co-chairing this session with Dr. Schmitz. We're going to go through as many of these questions as we can. Why don't I start off with a couple? We were looking at some of these questions and trying to group these thematically. But let's start with some of our first speakers, Dr. McQuade, Dr. Norian, and I'll sort of throw out the first question in a biased fashion because it comes from me. I'm curious, you've presented some really fascinating data with regard to modulation of immune cell populations in the tumor microenvironment, as well as interaction with tumor and the impact of obesity. I'm curious as to whether or not you've been able to look at whether obesity-associated fibrosis or changes in the extracellular matrix have any sort of physical properties that are altering trafficking of immune cells, or the ability of even some of these immunotherapies to be efficacious because of some of that extracellular matrix fibrotic deposition that occurs in the setting of obesity. I think either of you would be fantastic to address this question.
I can start if you want, Jennifer. So we have not looked at fibrosis specifically in our model, but I can tell you it doesn't seem like we've seen a lot of it in our kidneys from the obese mice versus, say, the obese resistant mice or the lean mice that respond to therapy. And in our renal cancer patients, we haven't looked at it at all, so I don't know. But I can say that in our mirroring model, both mice that respond to therapy, both obese and lean mice that respond to therapy, they actually have similar frequencies of infiltrating CD8 T cells that are activated. So unless there's some difference in the obese mice, the responders versus the non-responders, which we haven't seen yet, but we haven't looked at it really closely. So I don't think it's that, not at least in our model.
Can speak to, so from a melanoma perspective, I think it's a little bit interesting, right? That where we would think about the extracellular matrix and kind of immune trafficking would really be with the primary melanoma in the skin, which of course we aren't treating those patients with immunotherapy. I'm not sure that, that an obese state would have a different, a more fibrotic extracellular matrix, say, in a liver metastasis or a lung metastasis. Interestingly, though, the one area where this has been studied is not obesity, but age. So primary melanomas in aged skin do have a more fibrotic extracellular matrix, and Ashi Weeraratne's group at Hopkins has shown that in fact does influence immune cell trafficking. Now, with obesity and melanoma, obesity is associated with worse outcomes and an increased rate of metastasis in primary melanomas. So there it would actually be quite interesting to look at, right? Could that be one of the mechanisms?
Absolutely. Thank you. That's very interesting. We'll use, I guess, the chair's privilege to ask one more question. This one came from Katie. Katie, why don't I pass it over to you? I think you have the first question.
So yeah, this is again for our first two speakers. And I was just interested because there appeared to be a real focus on obesity with a focus on the adipose tissue and what the adipose tissue was likely doing. And so I'm curious to ask both of you whether you have looked at what the muscle tissue is doing, and given the likelihood that in these animals that you're studying that there is some variability with regard to the amount of and quality of lean tissue. Have you looked at that, and does it make a difference in any of your findings?
So I can start. So we have been very interested in this issue. We have not looked at this in our renal cancer patients, but we have plans to collaborate with an MD at UAB, whose name is Grant Williams. He published a paper in JITC in 2020 with Doug Johnson from Vanderbilt, and they actually looked at melanoma patients and ICI outcomes, and they found that patients who had the highest total adiposity tissue index, so they were doing this by CT scan, right, which is a much better method to use than BMI, but they found that patients who had the highest total adiposity tissue index, as well as low skeletal muscle gauge, indicating that they might have sarcopenia or loss of lean muscle mass, they actually did the worst. So there is quite a bit of evidence out there, and Betty Kahn has published on that as well. That loss of lean muscle is actually detrimental. We have not been able to look at that yet in our renal cancer patients, but it's something we plan to do. And in our mice, we have done body composition analysis, and we know that from the time of tumor challenge, so even if the mice start out obese, as the tumors progress, they lose weight. But interestingly, in our model, the weight that they lose is primarily their fat mass, which brings up all sorts of interesting questions, right? About, so the therapy is administered early on for us when the mice are obese, but then as the tumors progress, especially in the mice that aren't responding to therapy or in the mice that have no therapy, they have this very striking loss of quite a bit of their fat mass, but not their lean mass. So in our mouse model, we haven't been able to really study that yet because we haven't seen a big variability in terms of retention of lean muscle mass and loss of lean muscle mass, but it's something we definitely want to look at in our renal cancer patients.
And I think the other part that's important too for these studies with body composition analysis is that that paper that Lyse was just mentioning, one of the things is that those studies were done in the era where immunotherapy was not first line, and so patients were heavily pretreated, which could also influence those outcomes. So I do think it's important actually to look at this in frontline immunotherapy treated patients, and specifically things like melanoma and renal cell carcinoma, where you don't see a lot of cachexia. I would certainly, expect there to be cachexia, negative outcomes in, say, lung cancer patients treated with immunotherapy. And then I think as well incorporating, the other markers of kind of metabolic health also. I guess another question too, just for you to kind of throw it back to you and Ricky, Katherine, would be, when we do these body composition analyses, I've never really seen anybody where they actually also integrate questions around physical activity and exercise, as kind of a confounder, and how do you parse out, that overall health, physical activity, and then the positive effects that physical activity could have on the immune system versus, the actual muscle mass itself, which I guess we could do in mouse models.
Well, that's an excellent question. And I think actually Ricky's work is, sort of a precursor to that. And if I can take the chair's prerogative, Neil, and switch to a quick question to Ricky that isn't even on our list. And that is, it's sort of a corollary to a question that's on the list, Ricky, and that was, are you looking primarily at aerobic exercise? And I think you are. And so I think that one answer to Dr. McQuade's question is, I'm interested to see what your graphs look like if you add a resistance training group to some of your trials. And have you considered doing that?
Yeah, Katie, this is a great question. So the reason we're focusing on aerobic exercise is because we're largely interested in the immune cell mobilization and redistribution piece, which is largely driven by catecholamine signaling. So you get a much better catecholamine response if you put people on a treadmill or an exercise bike than you do if you get them to do strength training. But on the other side, I did mention that the myokines that are released during exercise also play an important role, and we do see a more robust myokine response after resistance exercise. So there's definitely scope for both modalities of exercise to play a role here. We haven't been looking specifically at resistance exercise in the individuals that have participated in our studies so far, but it's definitely something we have to do, absolutely.
I'll sort of dovetail off of that question and ask both Ricky and Katherine. I think that listening to both of your talks has gotten me, and I think several others listening, to think about the sort of the synergy of interventions that manipulate gut microbiome as well as immune response systemically, with potentially with exercise combined with diet interventions. My question, maybe for Ricky first, is, you talked very elegantly about cell therapies, adoptive cell therapies, as well as the issues with graft versus host disease, which of course is a major issue. Have you thought about ways to augment the therapeutic index of these therapies with exercise, and potentially using diet as an intervention to mitigate the GVHD while also potentiating or augmenting the therapeutic index of the cell therapy?
Yeah, so we haven't looked at diet so much in my lab. We are interested in exercise after transplantation to see what type of impact that can have. And we have been doing that currently using mouse models. So the data that I presented today was quite simply a passive infusion of resting lymphocytes versus exercise mobilized lymphocytes. But we have also looked at whether or not exercise in the recipient mice makes a difference, and we find that it actually does. So if you transplant human immune cells to the NSG mice, and then you get the NSG mice to exercise, the graft versus host disease is actually enhanced, which would make you think, okay, that's not a good thing. But whenever you have more graft versus host disease, you also have more graft versus leukemia. So whenever the mouses burn a tumor, then the anti-tumor response is actually enhanced. And then of course there's drugs that can be given to control graft versus host disease, like cyclophosphamide and so forth. So we still think that that's advantageous. But what is interesting in that model that we're using is we are looking at the effects of exercise training in a completely immunocompromised mouse model. And even when we don't transfer human immune cells, when we just give human cancer cells, there's still a positive effect of exercise, which lets us know that something is happening even independently of the immune system. And that's created a whole range of new questions in my lab that we really want to try and investigate.
Great, very interesting. Let me just have a look here. Katie, do you have one, or should we?
Sure. So this is a question for Dr. McQuade from an audience member. Are any of the IGFR antibodies helpful in your model? I know there are some companies that haven't found any synergy with these IGFR targets.
Sorry, so those have been tested in these murine melanoma models with good effects. Not specifically something that we've looked at in the context of obesity, but agree that clinical translation of the IGFR and the R inhibitors has been less than ideal.
So we were talking a little bit about uncoupling obesity versus physical activity. This question came in for Lyse, and the question is, do the obesity responders have elevated inflammatory markers? Do you have any review or data on participant level of physical activity that are still obese and how they respond to immunotherapy?
Well, that's a great question. So we do not have any data yet on physical activity, certainly not in our renal cancer patients. And I don't know, Jennifer, if you've seen anything about that, physical activity influencing ICI outcomes in cancer patients. I have not seen anything. I think this is I think it's a great area of research that we should start looking at, but we haven't seen that. I haven't seen anything really published on that. And then in the first part of the question, so for the obese mice, the ones that responded to therapy, they had downregulated the inflammatory pathways. So if we look early on at day 15, we see that in general, the obese animals have increased this IL-1 beta mediated signature, right, with the increased CXCL1 and increased CCL2. But if we look later on at individual mice that responded to therapy versus those that did not respond to therapy, the obese mice that responded to therapy, they don't have that inflammatory signature. So we don't know why that is, but for some reason, the responders are able to kind of down regulate or dampen that, pro-tumorigenic, pro-myeloid inflammatory signature. So it's not there.
There have been at least one study I know, and I think one that's still under review as far as positive effects of exercise on checkpoint inhibitor immunotherapy in mice. I have not yet seen any human data. We did take a really early look at kind of goat and leisure time activity and immunotherapy response when we still only had probably 100 patients or so in our database, and kind of saw one of those, distributions that you think of, right, where it exactly. So we're expanding that cohort now, and we'll be taking a look at that and also putting another context of diet and BMI and the microbiome.
Well, I have to say, as I listened to Katherine, I really kept wanting to have, and then listening to Ricky, I'm like, oh, I just want you all to collaborate together. Did you look at microbiome and diet-induced obesity and exercise? That's not a problem, is it? So coming back to a more answerable question, Lyse, this was a methodologic question specifically for you. There was a question from an audience member about whether the diet-induced obesity diet was just high fat or also high calories, and what the protein carb composition of obesity-inducing diets were in your animals.
Oh, sure. So the diet that we use is the research diets, 12492. So they have their formula on their website. It also does have higher kilocalories. It's a higher energy diet than a standard rodent chow, which is why in our model, we found that it was really important to use those obese resistant mice as a control, because they are fed that same RD12492 diet, but they are just resistant to becoming obese. So we have mice that are on the same diet, right, that we define as being obese and obese resistant. And if we do body composition analysis on those, we can see that the obese resistant mice really have body composition in terms of fat and lean mass that look very much like the lean mice that are on standard chow. But yes, it is a high energy diet compared to a diet that most rodents would be on.
I think there's another question from you that actually comes from Dr. McQuade, if I'm not mistaken. Is it from you?
Yes, it is. Yeah, and I can ask it myself.
Yeah.
So Lisa, I think the data that you showed is just beautiful, and it's so relevant of how you look at, dividing out the obese and then the control and then the responders versus the non-responders to immunotherapy. And I think it's a real challenge when we do these, when we do these experiments in the mice with immunotherapy, you have the responders and the non-responders, very different, right, than when we give them chemotherapy and targeted therapy. Everybody kind of responds the same. But this is what happens in humans, right? And so the way you divided it out is really interesting. My question is that, have you been able to take that, the human immune profiling data that you had done and divide that into those kind of 4 quadrants?
So we haven't. I think someone needs to do that. Our cohort is very much smaller than your cohorts have been. So, because we were just looking at renal cancer patients who received therapy, as standard of care. And so we have it from 2 different centers combined, but really we would need to have a much larger, I think, cohort of patients so we could subdivide everything out. That is something that we would love to do in the future, but we haven't done it yet. So I can't answer, but I do agree with you. I think it's really important because a lot of the studies that have been published so far on this issue, and as I said, I mean, we're guilty of it. Most people are. If you start by doing a retrospective study. So what do you have? You have BMI, right? But so I think it would be really important to try to do some prospective studies, maybe so that we could get longitudinal information on people in terms of their body composition. How is that changing over time? More information about diets that they're on, certainly more information about microbiome and how those profiles might be changing over time, right? So I think there's a real need to do some prospective studies so that we can gather that information over time. And then when we get to the point where we can define responders and non-responders from people who started out as non-obese or people who started out with obesity, we can then go back and identify some of those changes that were there maybe early on that are linked to therapy resistance versus therapy sensitivity in those 4 quadrants. I think that would be fantastic to do.
Yeah. And I'm just thinking back to, because again, a lot of my cohorts were actually not in the setting of, the biological ones were not in the setting of immunotherapy. They're kind of, older cohorts. And so then though, we think about the fact, right? So if the obese versus non-obese kind of have intrinsically different tumors, right? Like they have different metabolic phenotypes, then one would think that the patterns, the mechanisms of resistance to immunotherapy would be different, kind of based on their metabolic phenotypes. I think that's really cool. Can I ask one follow-up question? Which is, what was the mechanism of resistance in the lean?
Oh, yes. Okay. So I glossed over that totally because we were really surprised by that. They were such an outlier, on our hierarchical clustering and our heat map. So, and we haven't followed up on it because frankly, a lot of the markers are things that I don't know what to do with. So we like the highest change there in terms of genes that were upregulated is one that's called DEFB1. And
I had to look it up. I didn't even know what it was. So it's an antimicrobial. So, you know, Catherine's laughing down there, but so maybe you're more familiar with that than I am. But so I think it could be indicating to me that there's something in terms of a microbiota difference that was really driving the lean non-responders, and there are very few of them. I mean, but to make them be a lean non-responder. But yes, the highest upregulated gene in that cohort is DEFB1. So do with that what you will.
Lisa, I want to go back to this comment that you made. Actually, let me pause there before shifting the subject. Katherine, I think you were going to make a follow-up comment.
Oh yeah, there is some really nice data, at least in the melanoma field, that show use of antibiotics right before treatment actually reduces response. So the fact that you might see an increase in an antimicrobial agent in the non-responders is kind of fitting with the literature.
Thank you very much. We will look at that. Makes a lot of sense.
So, Lisa, to follow up on a comment you made, I think this sort of longitudinal tracking of changes in body composition is certainly an important issue that we discuss. What I've also been thinking about is particularly in immunogenic tumors, as patients progress through the natural history and lines of therapy, and potentially tumor mutational burden changes, neoantigen response changes, what I wonder is if there's an interplay between not only changing body composition, changing muscle mass, but also in intrinsic tumor response to immunotherapy, which I guess one might hypothesize would be augmented in later lines. You and Jennifer would know better than I, but have there been any analyses that have stratified by pretreatment or lines of therapy, and how that may intersect with the impact of obesity, even something as simple as BMI?
So, oh gosh, I can't think of the reference. I know that this has been looked at. And I do think it's an important question. So is it first line of therapy or is it subsequent line of therapy, when the immunotherapy was administered? I do, oh boy, can't come up with the reference, but I do know that there has been a paper published on this, and they did find that for immunotherapy given as frontline versus subsequent lines, they were seeing differences in progression-free survival and overall survival with BMI. So one way, I think it was immunotherapy as second line was beneficial, and frontline it was not beneficial with higher body mass index, but it could have been the reverse, and I'm sorry, but I know they were seeing different things, different trends, depending on whether or not the immunotherapy was administered as frontline or subsequent. So I think that's a really important question that we need to pay more attention to in the field as a whole.
Yeah, I appreciate that, especially as we start to think about intervening and at what point or where we want to design, in what setting we want to design some of these intervention trials. I'm also a little bit biased as a breast cancer person. I start to think about, up in the upfront setting, we really have hormonally driven tumors, but perhaps as we start to get into the more pre-treated setting, some of the immune-mediated pathways start to take over in terms of tumor behavior. So, I think this is a really interesting conversation that we're having here. Okay, let's go back to the list of questions here.
So we have two for Katherine that I think are very practical. So I'm going to pose them both together. They're to me, they're practical and related. One is, do large hospitals where patients seek treatment typically test the microbiome for bacterial strains? Is this considered to be part of best practices at this point before starting treatment? And I think that it's related to ask, do you see potential benefit for breast or any other type of cancer patients for adapting an anti-inflammatory diet prior to starting treatment to optimize their microbiome? So can you talk about sort of the practicalities?
Sure. First of all, I would love collecting fecal samples for standard of care in hospitals, because that would just allow us to do so much more kind of powerful kind of analysis. Right now, we're only really collecting fecal samples as part of clinical trials and with people who have specific interest in looking at microbiota regulated mechanisms. So currently, no, it would be fantastic if it was. And it's also important to note that an individual's microbiome is thought to be as unique as a fingerprint. So we really do kind of need very large studies to see kind of specific changes within gut microbiota populations. And then the second part of that question was
whether you advocate for an anti-inflammatory diet among cancer patients before they start their treatments in order to kind of prime their microbiota.
So I think this is going to be very context specific as well. You might not want an anti-inflammatory microbiome in cases where you want to elicit an immune response for your cancer outcomes. So there's going to be a huge difference in looking at prevention versus treatment. There's also going to be a very big difference in what types of therapies you are actually giving and the types of cancers. So what might be good for one context is not going to be the same for a different context. And so it's going to be very specific, and we're going to have to do a lot more research to actually determine which diet would be beneficial in which circumstance.
So I'm again going to take the chair's prerogative and just do a follow-up question here. I'm curious to get you to wax poetic just a little bit, Dr. Cook, about how far are we from the potential to use microbiome data clinically? Where are we with regard to, and I'd love to hear Dr. McQuade talk about this as well. Where are we? Are we still kind of looking at these huge plots and going, I kind of wonder what that means? Where are we? How far are we? Are there instances where we're close?
I would definitely say yes. There's been those two very recent studies that were published this year that showed fecal microbiota transplants in patients that increased PD-1 response in melanoma. They're fantastic studies. And I think using fecal microbiota transplants in a very controlled instance is kind of a more preferential and beneficial way of changing the microbiome. Things like probiotics that are available over the counter, there are only 1 or 2 different species. So when you give this oral probiotic, you are actually, in most cases, decreasing the overall diversity of the gut microbiome, which is not always a healthy thing to do. And so there is certain instances where having more of a population change using fecal microbiota transplants would be preferable over using 1 or 2 different species.
I feel like we have a session tomorrow called "What Can We Tell Our Patients?" and I feel like we need a little section on the microbiome here.
That's true, because there's also very much geographical locational differences in your gut microbiome population. So you see studies that happened in France are very different from the populations identified in Texas or Chicago. And so that also has to do with geographical locations, urbanization, diet, you name it, and it can modulate your gut microbiome. And so it's really important, one, that we get larger studies, but 2, try to control them as much as possible. So for example, there is sex differences, male-female microbiome. So if you're looking at a population, I do breast cancer, so obviously we just look at women, but it's important to also note menopausal status when it comes to that, because that can shift your gut microbiome. If you're looking at obesity, try to pick a cohort where they don't have comorbidities and are on other medications, because people have shown metformin can have huge effects on your gut microbiome. People have shown hypertension drugs can have effects on your gut microbiome. So trying to control it as much as possible helps us kind of focus on which bugs and populations could be causative rather than correlative.
Thank you.
Do you have a
Jennifer?
sense of the, oh, I'm sorry, I'm just going to butt in real quick for
Go ahead.
Katherine to follow up. Do you have a sense of the effect size or at least in your mouse models, of what dietary manipulation, getting back to the original question that Katie posed? I'm always curious as to, we do hear a lot about how medical interventions can vastly manipulate the microbiome. Does the effect size of dietary manipulation compare to what you're observing with regard to some of the associations you just raised?
So in our very controlled preclinical mouse studies, diet is the #1 influencer on the gut microbiome, and shifting the diet will give you the biggest results. Giving oral probiotics, doing some exercise, increasing fiber moderately. These approaches can cause shifts. Giving anti-estrogen drugs, giving immunotherapies have very small shifts within dietary populations. They are significant, but diet is the #1 modulator.
Fascinating. Dr. McQuade?
Jennifer?
Yeah, I was just going to go back. I think that there's two real components to this. And so one is looking at the microbiome as a biological predictor, as a biomarker. And the second is, are we ready for it as a target? And so as a biomarker, I think we've still got a ways to go. I think what the original questioner was kind of getting at is, can all of our patients, can we, clinically run microbiome profiles and then say, you have a good or a bad microbiome, and therefore this is what you should do. And I think that we're just not there. There's still a lot to understand about what really is kind of a good versus a bad microbiome. And with all the stuff that Katherine was mentioning, as far as, the geographic differences, the fact that a lot of these studies, kind of the pro-response taxa are not necessarily overlapping, right? And so is it more about function instead of phylogeny of the microbiota profiles? The second, as far as, then are we ready to start modulating the microbiome? And in a research context, absolutely, the FMT stuff, as Katherine said, is very exciting. Our work has been on looking at the impact of dietary fiber on the microbiome in response to immunotherapy, with a paper forthcoming. But there we actually, I'll just put in a plug for my lab has a poster here as well, looking at our studies of a high fiber diet controlled intervention on modulating the gut microbiome and trying to enhance response to immunotherapy. So I think it's coming, but it's also that question of how do we select who's best for a diet versus for an FMT, or do we need a symbiotic? Can we kind of have rational designed multi-strain probiotics, since the single strain ones are not the answer. So there's a ton still left to learn.
So there's a question that just came in. We actually have some questions for Ricky, but a question just came in that I think is really pertinent to the discussion we've just been having, that I think is probably for all of you, and that would be, would small studies or even pilot studies potentially help to give direction to informing the types of larger studies that we've just been discussing? Because the larger studies will take a long time to mount and to get funded and to get the results. So is there anything, can any of you speak to, okay, so how would you do the smaller study to decide, whether we should be giving a particular diet or the symbiotic or what are those smaller studies? How do we try to approach this without needing to have giant long trials?
So yeah, we've tried to take the, kind of the drug approach model, right, of 3-phase studies. And so our phase 1 was feasibility. And here again, we're using controlled feeding intervention. So I want to know, when I kind of take out that behavioral aspect, and if I give patients all their food, can I actually modulate the microbiome? And so that's the study we just completed. And then phase 2 is now, in the setting of immunotherapy. We can then start to learn how are we modulating the microbiome, and then does it impact immune response? But then the third, ultimately, can we influence outcomes? Those have to be big studies. The question is, if it's something like a high fiber diet, I don't really have any problems telling my patients to do that now. I just don't have the data telling them that it's going to make a difference to their immunotherapy response beyond our retrospective cohort and mouse data.
Katherine, any comment on how to do something that isn't a giant study to try to answer some of our lingering questions about the microbiome?
At least I can address some of the preclinical concerns using smaller studies is try to make them as translationally relevant as possible. Use dietary components that make sense, use drug doses that make sense, and use combinations that make sense. Because all of these things are going to affect the gut microbiome, and you might come to a result at your endpoint that won't translate into a human population if you don't use these translationally relevant kind of parameters.
Great, very interesting. So we do have, as Katie mentioned, we do have some questions, Ricky, for you. This question comes from Karen Collins, and she's asking, do the studies involving exercise above a lactate producing level imply that vigorous exercise is needed for these effects? In other words, is moderate walking enough to see some of the effects that you were describing?
Yeah, it's an excellent question. What we've found is that the exercise has to be intense enough to evoke a catecholamine response. So by definition, that would be considered vigorous exercise. But data did show today, after we made the observation that blocking the beta-1 adrenergic receptor can actually boost the response, we decided to repeat that study, but looking at different intensities of exercise. And what we actually found in healthy individuals is that if we gave them bisoprolol, so that's a selective beta-1 antagonist, and we exercise them at 50% VO2 max, which is not really vigorous exercise at all, the numbers of natural killer cells that were mobilized into the blood at 50% VO2 max with bisoprolol were about the same as what they were under a placebo at 75 or 80% VO2 max. So it's almost like we can manipulate adrenergic signaling to amplify the exercise response at much lower intensities of exercise. So that is a good question. It's something that we're focused on.
It's really fascinating. It almost sounds like there's this sort of stochastic response between, beta adrenergic blockade and what you're seeing with exercise response. And I'll go back to a similar question I asked about effect size for other interventions, but do you see a similar response to, let's say, vigorous exercise versus beta-1 blockade?
So Neil, just to clarify, just looking at vigorous exercise compared to beta-1 blockade at lower intensities.
Right.
Yeah, that's exactly what we see. So, and if we just give a placebo and we exercise people at 80% VO2 max, the number of cells that are mobilized is about the same as what we see at much lower intensities of exercise when we block the beta-1 adrenergic receptor. And then we've been doing other things with phosphodiesterase inhibitors, to try and manipulate that pathway further. We've looked at some so-called beta blockers that have biased agonist activity. So you take carvedilol, for example, which blocks GS signaling but actually augments beta arrestin signaling. And that's why we really want to try and unravel all the downstream signaling pathways to really find out which drugs might be beneficial for cancer patients.
So the last question for you, Ricky, is, and I kind of want to pose it if I can jump in, Neil. I want to ask because I want to pose it in a slightly different way. I'll say what it is, and then I want to repose it. In patients who are unable to exercise, would external muscle stimulation have similar effects to exercise? And I'll let you answer that, but I'll ask you, and you've gotten to this a little bit in your work about exercise mimetics. It's an issue that comes up all the time if we're talking about trying to see what exercise can do. You know, are you chasing any particular exercise mimetics?
You know, we're not specifically at the moment, but you know, to answer the first question, and I'm having to go back a long time ago, but when I was at the University of Houston, we did do a study with tetraplegics where we used forced muscle stimulation exercise in these individuals. So essentially, you can attach electrodes to the muscles, and then you can get them to operate a cycling ergometer. And when we did that, we did see a mobilization of immune cells into the blood. It was relatively small compared to able-bodied individuals doing exercise, but it was something that intrigued us at the time. I don't know if other groups are looking at just forced muscle stimulation in more detail. It's not something that we're looking at in our lab.
Okay, I think we're nearing the end of this session. Let me see if we have any pressing unaddressed questions here. I think we actually got through all of them. Katie, did I miss any?
Yes, there's one that is about usefulness of fecal samples. Could you speak to the usefulness of fecal samples versus colon biopsies or other methods for evaluating the gut microbiome in humans, with consideration that fecal microbes are what's coming out of rather than what's being retained by the body? I believe that's for Katherine.
Yeah, there is definitely going to be shifts in the proportional abundance of microbes depending upon where you're sampling within the intestinal system, also what's being excreted in fecal samples. This has a lot to do with just geographical location, pH, hypoxia levels. Those are all gradients within your GI, and that's going to cause a selection of different types of bugs and allows certain ones to grow more favorably in the colon versus the descending colon versus the small intestines. And so where you sample does influence your results. And I think that measuring what's excreted versus what's present, there's definitely going to be a difference. There's also that's going to change your results. And I don't know if I can attest to how much it's going to change the results and whether or not this is just due to a sampling feasibility and ease. Obviously, it's a lot easier to collect fecal samples after they're excreted. You don't have to go in there and go get them. But I do not actually, I don't think I can actually tell you whether or not studies will cause changes depending upon, and what those changes means depends upon where you collect them.
Great, okay. Well, I think we're going to wrap it up here. That was really a fantastic discussion. Thank you very much to all of our speakers. Thank you very much to our co-chair. And thank you to all of you for listening and submitting these really, really fantastic questions. We look forward to continuing this discussion in the subsequent sessions this afternoon. And also tomorrow as well. As you heard from Katie, we have a session wrapping things up tomorrow, talking about what can we tell our patients. So please continue to ask these fantastic questions so that we may ultimately bring this data to our patients. Thank you very much.
Thanks, everybody. Bye.
Bye.